2023 Biology Head Start lecture. I hope you guys are all doing really well and are able to hear me okay. So welcome. I hope you've had a good year so far, a good start, a solid start to 2023. This year's probably, full of adventures for you with finishing high school and then continuing on. So it's exciting stuff happening. So welcome. My name is Aditi and today I'll be taking you through our lecture. So firstly, like I said, I want to welcome you all from all of us here at ATAR Notes to this lecture today. And as you know, ATAR Notes here, we're all about helping you guys achieve your very best. So a very warm welcome to you. Now, before obviously I go on, before getting into content today, I quickly do want to talk a bit about what resources we have to offer and how you can use ATAR Notes to optimize your study this year. So we have lots of free resources that you are able to access through our website at atarnotes.com. And you can register and then be able to download all of these resources. We've got study notes that are put up by your peers, and you're able to access other people's notes, add them and collate them to your own notes. So it's quite helpful to look at how other people have looked at a different subject and being able to add to your own knowledge. We've obviously got our lectures on this week, so definitely register for, there's no limit for how many you can register for. So definitely go ahead and register for all of the ones that are relevant to you because they're so helpful. And that's, coming from me as a past student who's been to these lectures, who's used these resources from ATAR Notes and found them super helpful. We also have online forums where you can obviously, you're able to have discussions with your peers on the forum as well, but also moderators from ATAR Notes who are, who jump on frequently to help you out, like for English especially. We're always like jumping on to talk about different texts, different questions, and same thing applies for like concepts in biology. So a huge range of different topics. And things that you can access there. Along with that, we've got videos, online revision videos that you can also access on our website, newsletters to help you stay in loop as what's going on here. There is an ATAR calculator that's available. Not quite useful to you just yet, but you'll be getting there soon. So, it's a great place to sort of go, I don't know, put in your hypothesized marks and then, get a gauge where you are at and then being able to work towards it. But that's a resource there for you. Then we've got articles, study strategy and tips, and finally a lot more resources available for you. So those are our free resources that you can find at atarnotes.com. Along with that, we also have some paid resources that including TuteSmart, which is our tutoring facility. So with TuteSmart, it's basically online tutoring. I'm a tutor here at TuteSmart. I tutor English and PDHPE. So we've got tutoring available from recent Year 12 grads. So it's tailored to your needs. It's all about you. And that's our funder, that it's all about you. We are here to facilitate your learning. So definitely check that out, and I'll be talking you through some offers today as well, some sweet deals that you can utilize. We've got our study guide, so our printed revision materials that are written by recent grads that cover a range of different tips, techniques, and of course content that is based on the syllabus. So it's quite it's very useful. And an online version of that, which I kind of like to think of as a Netflix for all our study guides, is Unlimited. It is a personal favorite of mine because it's got all your study guides in one place. You can access all your guides for all different subjects, and along with that, there are also practice question guides available up there and text guides that are relevant for like English. So definitely have a look. And like I said, I'll be giving you some sweet deals here today. So before we actually go on and I introduce before we go on and I introduce myself, I kind of want to get to know you guys first. So today, and I'll talk you through this kind of later too, but today we'll be communicating through Slido. So Slido, we'll be talking to each other through there Please post lots of questions, upvote other people's questions. I've got Slido open at the side here, so if I look here towards my iPad, I'm looking at what questions are up on our Slido. So I'm just gonna come out and pop that up on here. So, oh, okay, there we go. Got the right Slido up for us, and okay, let's get started. So yeah, I'm going to open up my polls for today, which is to allow me to get to know you guys. I've got some awesome questions up there already. I'll come back to these in a moment. I'm going to jump to my polls first, and I'd like to know how we are all feeling today. How's your day been? That's 4 o'clock in Australia, so I'm overseas right now, so that's why if you see it's very sunny here, because I'm overseas, so I'm in a different time zone. I'm 5.5 hours behind you guys. So, okay, we're all feeling pretty good. Yeah, I put one on there because I know it just feels like the holidays flew by, didn't they? And I feel the same, honestly. At one point you're thinking, okay, I've got a solid month. I'm gonna catch up on my work. I'm gonna have a good holiday, and then it's like, oh no, it's halfway through already, and school's gonna start again. So we kind of, okay, so we're all getting there. It's pretty good with that sort of that back thought of, okay, school's gonna start again. Cool. All right, I've got some good questions coming up. I've got a cool. Okay, cool. I will do my introduction soon, so you'll get to know me soon. But yeah, I'm just gonna want to know you guys first. So awesome. So school's starting again. Yeah, get that one. And how was term 1 of Year 12 biology? How do we feel after that? It's definitely a bit of a jump from Year 11. So it can feel, that little bit of like a bit jarring once you get into it. But once you sort of start flowing through it, and with these next 3 terms, you'll find that because it's continuous, you're getting the gist of the content, it feel a lot easier. So yeah, so we are all like, okay, pretty good, better than I expected. That's awesome. That's so good to hear. And absolutely, awesome. I've got some absolutely amazing. I can't see what percentage it is because of this thingy here, but let me see if I can see it on here. Okay, well, we've got 7% feeling absolutely amazing. That's awesome. Good, better than you expected. Okay, that's really pleasing to hear. I'm glad you guys had a good term 1, and I assure you it's going to get even better from here. Okay, now, the fun stuff. What module did we cover last time? I know more than likely would be mod 5, but you never know. Like some schools start to, go out of order, and they may do cover another module first. Okay, so I've got someone who's done infectious disease already. Okay, cool. So today, okay, we've gone on module 8 as well. Okay, cool. So today we'll be going through recapping module 5 and then getting a head start on module 6. So okay, so far I've got no one who's done module 6, which is good because we're gonna have a head start in that. Okay, got 1%, cool, but 80% have done module 5. That's awesome. Okay, got my Q&A. All right, I've got some awesome questions coming up. Great. Okay, so most of us have done module 5. Some of us have gone ahead and done module 7 and then 8, and then 1% have done module 6. Cool. So today will be relevant for everyone because we'll be going through module 5 recap. If you've done it, it's going to be a bit of a refresher from the holidays. If you haven't done it, it's a head start. Same for module 6. So, all right, how do we feel about module 5? Okay, my Slido on the side keeps logging me out, so I'm going to put that in while you guys are answering the question. Okay, so basically I've got people filling in who haven't studied it yet, so I should have a lot more answers coming because 76% of us have done module 5. All right, there we go. So pretty good. Glad it's over. Yep. It can feel like there's a lot of content, and there is a lot of content that you need to go through, so I can totally understand that feeling. Absolute nightmare. Yeah, a lot of information going on, but hopefully today will help simplify that info. And by the time we get through, you will feel a lot more confident with it as well. Okay, sorry, give me 1 second. Okay, so my slide is opening up on there again. So, okay, cool. We're feeling pretty good with it, and we're going to be absolutely confident with it by the time we're done today. That's my goal. So, which module are you covering next term? Let's get to that one. What are we doing next term? Okay, cool. Yep. So most of us are doing module 6 so far. I'm going to wait for more answers to come in because it's just 20 people. So let's wait for the whole cohort and then we'll go on. But yeah, it looks like most of you guys are doing module 6 next term. Then we've got module 5 in the lead. So that's really good. Okay, I've got some amazing questions on the side too that I'm keeping an eye out on. There was one earlier, and I'll talk you through this soon too, about the resource and the slides from today. So you'll find the slides today on the portal through which you are accessing this stream currently. It would just be like below the stream, you'll find the slides for today. These are recorded, so the lectures are all recorded, so you'll be able to access this recording as soon as we are finished. Cool. So most of us are doing module 6 next term. I was going to say next year, and I was like, nope, it's 2023. So yeah, most of us are doing module 6 next term. So exciting stuff. Finally, what is our biggest, or second last question, what is our biggest concern with biology right now? What do we feel is, that little thing that's nagging us, that's really annoying? Everything. Yeah. That's a good one. Yep, fair. Remembering everything, absolutely. So much content for sure. Digesting all the content. Yep, memorization. Wording. Yep, answers. That's huge, right? Like being able to word your answers and be able to express yourself scientifically and succinctly in biology is quite important. So we'll talk about that. So much content, I know, I totally get it, guys. I so do. There is indeed a lot going on, but hopefully today will simplify some of that for you. Yep, assignments, so much content to remember, wording my answers, structuring answers, falling behind. That's a very fair worry too. So we'll talk about some tips at the end, some study tips and stuff that are going to help you with that. Lots of different things work for lots of different people. That's how I like to think about it. So I'll be talking you through some study techniques that work for me, and hopefully they work for you as well. But I'll be talking about a range of them. So maybe something will work for you. If something doesn't, experiment with them. And I'm sure you'll find something. Extended responses, that's a big one. Yes, remembering it all. Ranking, yeah, with ranking, that's a good worry to think about. From the get-go, is that you need to sort of stay in a specific spot to achieve your specific desired grade, your desired rank, and ultimately your desired ATAR. So can I just say from the get-go that what you do in class right now, your all school assignments, they are 50%. But the 50% that is, I think that's more important, is locked away until the HSC. So 50% of everything, of your grade is dependent on how you perform in the HSC. So even if you feel that term 1 assignment didn't go as well as you wanted it to, that's okay, because you obviously got 3 more assignments to go, including trials for the school assignment, and then you've got 50% locked away in a treasure chest for you that you can work towards. So it's looking quite optimistic from here. So I do want you to sort of think of it that way and think there's a lot that I can do right now. Okay. So I've got good questions coming through. Keep them coming, and I'm going to jump to them before we get started, but I do want to be mindful of the time as well. Last one for now. What are we hoping to get out of this lecture? What do you guys want me to focus on, right? I'm obviously going to go through the content, but I'm happy to spend a lot more time going through questions, talking through study tips. So if you've got questions about that, sort of topic, please put them up and upvote other people's questions. I can get to those burning questions first. Study tips, yep. How to study better for biology. Yep, totally get it. And that's a good place to start. How do you study in order to study effectively and then get your desired marks? Get ahead on some content, sure. Yep, insight on some concepts. Yep, ways to study, module 5 revision. Study tips, content. So those are our main how to attack those extended questions. Yep, we'll be going through some questions and breaking them together as well, breaking them down together. So don't worry, I've got some questions, revision questions up on the slider and on the slides. So you'll get a lot of practice today with that too. Yep, finishing content, yes. Okay, cool. Helpful notes, yep, so hopefully the slides will be helpful, and like I said earlier, I'll be going through and I'll be talking you through some of our resources today, including Edunlimited, which is an awesome resource. So hopefully you'll find that helpful and want to have a look at that too. Insight, yep, head start, deep understanding of modules for sure. I think once you get that deep understanding of why are we learning this, how does that apply to the real world, it makes the concept more interesting and more exciting to learn about. So hopefully I do want to do that today and get into some topics like that. So yeah, perfect. All right, let's move on then. What I'll do is I am going to come to these questions, but what I might do is I might just wait till the break because there's a lot to talk about here. And once I start talking, the thing is I keep on talking. So I don't want to run into content time just yet. So what I'll do is I'm going to wait for these, come back to them in a break, and then obviously at the end of the slides too. So keep putting questions up, and I want to spend a lot of time going through them. For now though, I'm going to jump back into our slides for today and get started. So I've got to know you all, what your worries are with biology, how you all feeling. So now so I'll introduce myself now. So like I said, I'm Aditi. I graduated in 2021, so that feels like ages ago, but at the same time it feels like not a day has passed. But anyways, I graduated in 2021 as the Ducks with an ATAR of 95 with band 6s in English Extension, Advanced PDHP in Biology. I am currently studying this kind of same field that we'll be going through in biology today. I'm studying Bachelor of Science and Bachelor of Advanced Studies, majoring in medical science and political economy at the University of Sydney as a Dell Scholar. And personally love reading, huge English nerd, good shows and sunny days. It is a sunny day where I'm here today. I hope you guys are also enjoying the sun. It's winters here though, so it's like freezing but sunny, which is good. But yeah, I hope you guys are all having good weather there too. I've heard there's been rain, but hopefully it's nicer now. So yeah, that's it from me. If you have any questions about any of my other subject like English or something, pop them up. If you get the time to come to them at the end, we may. If you've got questions about like the degree or any of the other like processes that are related to university or tertiary education, please pop them up. I'm happy to answer those as well. So there's a lot that you can ask today, so please go ahead, ask the questions that are relevant to you, and I'm going to try my very best to come to them. So now, without further ado, I am going to get to our questions and get to our content as well. I'm seeing some awesome questions coming through, so keep them coming, and then we'll get into it. So Module 5 recap, that's where we're going to start off today, going through the module. I obviously won't, because it's recap, I won't be going through every little point. I'm going through the main concepts within Module 5. You'll have the slides, and I'm going through like those really, juicy parts of the module where there's a lot to remember. So that's what I'm going to try and break down today. Then with Module 6, we'll be going through the whole module. I've got all the information sorted out according to the syllabus dot points. So hopefully it's nice and easy for you to then, once this lecture is done, to then go through on your own and have a look at, and obviously use it as a reference, for your studies. Then we'll be having a proper Q&A at the end, where we go through, all your questions. So please ask as many questions as you like, and upvote other people's questions. That's really important. Like I said, we're communicating through a slider, but you're very familiar with it now. So keep asking questions. If there are any content specific ones, go for it. But yeah, like it's up to you. Whatever you want to ask, I don't mind. Okay, so now let's get started with Module 5 Heredity. A lot going on here, so let's break it down first. As you would know by now, you are studying 4 modules this year, right, well, across year 12, because part of it was last year too. So you're studying 4 modules in year 12. It's a kind of a continuation on from year 11. So I did see some questions about, how relevant is year 11 content. Look, now here's the thing. Some of the stuff like, all the ecosystem stuff that you do at the end of, like, usually in term 3 of year, so that's module 3, I believe, in year 11 biology, like, all your not Punnett squares, all the measuring stuff, and you're looking at ecosystems and stuff, that is going to be relevant, but it's sort of relevant in theory, you're not good in theory. So, you're looking at, for instance, when you look at infectious diseases, you're looking at how they spread across environments, what is the effect of ecosystems on there. But what really is important, I guess, from year 11 biology will be repeated in year 12 biology. Like, it'd be a base, right? So it wouldn't be like, you looked at respiratory systems and stuff in module 2, so we are using parts of it in module 5, but it's not that the same content is being repeated. I apologize for the noise in the background. I've got some construction work going on, so I apologize. Hopefully it'll stop soon. But anyway, so what's really important will be repeated, like your teacher will go through it, you will come across it, but there isn't anything that's sort of directly taken and put in there that you wouldn't know about. There are going to be concepts that you apply, but you come across them in year 12 biology too. Yeah, and year 11 biology is a base. Like, you learn the concepts, for instance, in module 2, you learn the different parts of the human body, you learn different systems in small depth. But then when we look at module 5, you're going into more detail with, like, the reproductive systems, with how reproduction is done and how that then affects, generations and living beings and stuff. So you're kind of taking that little point and going off, exploring them more in a lot more depth. That's what I mean. So don't get too worried about, if you, let's say, if for instance year 11 didn't go as you hoped and you thought, okay, year 11 biology didn't do as well, I'm scared it's going to affect my year 12 biology. Well, if you don't like it, then it will. If you don't like biology, you're like, I don't want to do this, but I have to do this for my degree or whatever, it will. But if you sort of think at it from a perspective that I'm gonna learn new things, it's not going to be exactly what it was in year 11. It's going to be new stuff. I personally like year 12 biology more than I did year 11 because there was a lot more going on. Like, you were looking at the human body a lot more. You were looking at organisms a lot more, and how like DNA and stuff works. I found that a lot more interesting. So give it a go before you go, all right, not my thing, don't want to do it. So definitely give it a go. But that's what. But getting back to my point, we've got 4 modules. We are starting off with module 5, heredity. We'll be looking at reproduction, cell replication. You would have looked at, like, parts of the cell back in year 11. Now you're looking at a lot more depth as to what's going on within all of these different cell parts, and then how and specifically look at that in light of DNA, replication. So cell replication, DNA and polypeptide synthesis, genetic variation. So we start off by looking at these specific, bodily processes, and then we're zooming out to see what impact is that going to have on the environment, and on what impact is that going to have on the environment. So from there, I apologize for the noise again. Hopefully you're all able to hear me. I'll speak a bit louder. So from there, we've got module 6, genetic change, where we look at mutations, we look at biotechnology and genetic technologies. We won't be covering module 7 and 8 today, but that will be in next lecture series. And you're also able to access our old lecture series too. So if you are interested in these, go ahead, access the last year's one, and you'll be able to have a head start on these too. Like, that's personally work for me too, where go back, you have a look at the older lectures, and are able to access that content too. But for today, we'll be looking at module 6 and module 5. So yep, that's module 6. So let's start off with that very first question. how does reproduction ensure the continuity of a species? It's got two parts here. Firstly, how is that happening? How is reproduction happening? Secondly, how is that ensuring, how is that going through living organisms and generations in order to achieve the continuity of a species? That's what we are looking at. So we'll be looking at reproduction, like I said, cell replication, DNA and polypeptide synthesis, genetic variation, and inheritance patterns in a population. There's a lot going on here, a lot of info. So I'm going to try and go through the theory and break it down for you. If you've got any questions, please pop them up on Slido. So firstly, we'll be looking at how traits are passed from generation to generation on a system, cellular, and molecular level. So like I said, we're looking at those specific, cellular processes and then zooming out to look at how's that showing up, how is that affecting the whole ecosystem. Then we'll be looking at how genetic information stored as DNA is expressed in external traits, how variation occurs in populations, and how this variation may be traced within populations. So there's a lot going on here, and like I said, I'll be breaking it down for you so you feel confident with these concepts. And as you go through the lecture today, please take some notes. It's going to help jog your memory and ensure that you guys are, focused. Sometimes you're listening and then you zoom out. Totally done that. So try and make mind maps. Mind maps are a great way to, connect things. So make mind maps, for instance, on how these different things connect. And then by the end of the lecture, you'll have some notes ready for you to go that you can use in your revision this year. So we'll start off by looking at, in the syllabus, they want us to look at animals, so they want us to look at external fertilization and internal fertilization. What we're doing here is we're looking at the advantages and the disadvantages of both. With external fertilization, what are the advantages? That a species an organism is able to breed more, right? But what is the disadvantage? That they breed more because not all of those gametes will actually fertilize. So that's that's a disadvantage of that, along with the fact that even after fertilization occurs, we are not sure if we're not sure about the safety of that just like the word just oh my goodness of the blastocyst, which then becomes the mammal. So we're not sure about the safety of it either. Then we've got internal fertilization. A disadvantage is that it's limited in the sense that you may not be able to breed as many organisms, but advantage is that there's the safety of the safety, and fertilization it will occur more. There's a high percentage of it occurring in comparison to external fertilization. Then we've got plants, where we're looking at asexual reproduction. So we look at these different methods like vegetative propagation, apoptosis, and we're also looking at sexual reproduction like pollination and stuff. Then we've got fungi, so we'll be looking at, so we are discussing asexual reproduction, the different processes. So we're looking at budding, we're looking at spores, and also fragmentation. So you want to have examples for these. And then we're also looking at sexual reproduction. In protists, we are looking at asexual reproduction, so binary fission, right? And then budding. How does binary fission occur? It's when that one cell replicates into two. Then we've got sexual reproduction too that we'll be looking at. And finally, in bacteria, you're looking at binary fission specifically. For all of these, you want to have examples. Like I said earlier, I won't be going through breaking each one down, but I am sort of talking you through each, so hopefully you've got examples ready to go. And if you want to ask more specific questions to this, please pop them up on the slider, and then I can talk through the concepts more. But I'm just giving you a general overview of this. But I will talk a bit more about asexual versus sexual reproduction and what are the benefits and the disadvantages of each, because that's relevant for all of them. Even though for animals, it isn't said that you're still looking at asexual, you're looking at asexual reproduction versus sexual reproduction, and you want to have examples like for animals that do asexual reproduction, even though most of the mammals will be doing sexual reproduction. So just gonna have a quick drink. So asexual reproduction, it's quick, right? It's quick. It does not require mates. It's not energy intensive, and there is no requirement for offspring care. And that kind of then, seeps into like even external fertilization, where we are not worried about the we're not worried about the offspring. The offspring is supposed to make it on their own, but that's not to be confused here. What I mean is that that when you look at asexual reproduction and you're looking at, for instance, animals, you look at sea animals, so you can then go on marine animals, you can then go on and talk about that. But with sexual reproduction, there is the production of new alleles, so it's beneficial for biodiversity more than asexual reproduction because the offspring will just be a clone of the parent. There's also variation and the ability to adapt to the environment, so that links directly back to natural selection. With asexual reproduction, however, of course, we've got disadvantages, is that their clones lacks diversity. Reduced adaptive abilities. They're more susceptible to large-scale extinction events. So you'll also focus a bit on this part in Module 6, but I'll get to that when we get to that. I don't want to skip ahead, but I'll sort of come back to this when we talk a bit about Module 6. Then in sexual reproduction, disadvantages are it's time intensive, energy intensive, you need a mating partner, and there are fewer offsprings. Okay, so now let's have a look at fertilization. So as we know, fertilization is the fusion of gametes to initiate the development of a new organism. Implantation, so here is a more detailed diagram that goes through each one. So we've got fertilization, and then you've got early implantation where a fertilized egg adheres to the walls of the uterus, and at this point it's called a blastocyst. Then we've got hormonal control. So you're looking at the role of the endocrine system in controlling the pregnancy process. So you will specifically be looking at some hormones too. You'll be looking at, for example, oxytocin. You want to look at also, there's a bunch listed in the syllabus like testosterone. You want to look at pregnancy hormones specifically though. Testosterone is earlier on when we look at male and female hormones, but within hormonal control, pregnancy hormones, we're looking at oxytocin and the whole and there's a bunch of hormones that you kind of want to look at and have examples for. We then want to be able to have this knowledge, right, of how is fertilization occurring, and then be able to manipulate it in order to so we want to be able to manipulate the process in order to have a specific impact. And what I mean by that is selective breeding, right? So we want to be able to know the process, know how it can be manipulated in order to get our desired effect. In this case, maybe a specific, especially when this ties in a lot to Module 6 when we look at GMOs, we look at different there's also animal examples of genetic modification. But again, I'll wait till I get there. I do have a bit of a habit of skipping ahead, so not going to do that just yet. But an example here, like I said, is selective breeding. So we understand that in order to achieve pregnancy, the sperm needs to encounter the egg at a certain time. So we time breeding, right? So that it occurs during the intersecting fertility cycles here, 12 to 24 hours. And we know that the phenotypic traits are heritable. So if you're looking at, like for example, cows, let's say a specific cow has, it delivers more milk, or it's more meaty, for example, whatever the phenotypic traits are, they're heritable. We want to be able to extract those and perhaps mix that with another breed's heritable traits that are desirable, or we just specifically want those traits to show up in the phenotypic expression. So therefore, we are influencing the traits of livestock offspring, and we can crossbreed or we can purebreed. Either one works here, depending on what those desirable traits are. And this, of course, has led to the development of new species, for example, the Jersey or Angus cows. And here is an example there. So that's what this idea here of having this knowledge, being able to manipulate it, and then get a desirable effect. That's what's happening here with reproduction. But within the syllabus, you need to know about reproduction in mammals and fertilization in mammals, and then you specifically look at selective breeding later on too. So now we get into cell replication, which I'm sure most of you will agree there's a lot going on here. So I'm going to break this down, and the inquiry question here is, how important is it for genetic material to be replicated? Exactly. Um, it's a bit of a study tip, but once you sort of go through the modules, you should be able to come back and answer these inquiry questions in like a solid extended response using all the information that's listed underneath that dot point. So it's all about being able to come back and say, all right, I've learned these concepts, and this is how it's relevant to this question of how important it is for genetic material to be replicated exactly. So all of these processes then are doing this to ensure that genetic material is replicated exactly. So there's a lot going on here, but think of those concepts and think they all need to come back and link to this big question that you should be able to answer in like I said, an extended response, a 9-10 marker, a solid response. Okay, so cell replication, meiosis. So I'm getting straight into meiosis today, but as we know, there's mitosis and meiosis. Because we're looking at cell replication and genetic material being transferred, we're looking specifically at meiosis. So key structures that we want to look at: chromosomes, nuclear envelopes, a mitotic spindle, and the cell membrane. So as you can see here, we've got the homologous chromosomes. This middle part here is the centromere. So the chromosomes are counted by the centromere here. So this is one chromosome and this is one chromosome, but this is also one chromosome because there's that one centromere there. So okay, so like I said, so we've got that one chromosome here and another one here. It's a homologous pair of chromosomes, and then we are replicating it. So just here we're getting a copy of that chromosome, and same thing happens here. So these two are called sister chromatids, and they're both homologous chromosomes. So here we need to know rounds of division. How many rounds of division are happening in meiosis? That's 2. The number of daughter cells that you get, that's 4. So you end up with 4 daughter cells in meiosis, but in mitosis you end up with 2. The chromosome number of daughter cells, it's a haploid number, so it's going to be 23. And then the role in the body, why is it important? And that's what we're going to go through now. So I've got 2 diagrams here, and I'm going to simplify this for us, but we need to remember that meiosis is happening here. We've got meiosis 1 and meiosis 2. So, well, I mean, generally all the fun stuff happens in meiosis 1, all the crossing over and stuff, but you need to remember both of them, and you need to remember the process and the names. So prophase 1, so we start off with our interphase, prophase 1, metaphase, anaphase, telophase, and cytokinesis, and then we've got the same thing happening here. So what happens specifically? We start off with our centrosomes here, our chromosomes are lined up over here. Then that DNA is copied, it's replicated, right? And then those chromosomes are going to pair up. That's prophase 1. So interphase, we're getting ready for our DNA replication. The DNA is replicated, so we've got prophase. As you can see, the chromosomes pair up. Then recombination occurs. So actually, when our chromosomes pair up, and then recombination occurs, here we've got metaphase, but there's also pro-metaphase. So something to know, but we kind of go from prophase to metaphase. Again, sorry about that. But like I said, I'm going to speak a bit louder so hopefully you're able to hear me okay. So we've got chromosomes that line up at the equator. Sorry, give me 1 second. Okay. Alright, so we've got chromosomes that line up at the equator here, and we've got the mitotic spindle there. And then we see crossing over, so it happens over here, and then chromosomes are pulled apart. So over here you can see now crossing over has occurred. The genetic material has been exchanged. When they're crossing over, it's going to be the same. When they line up, it's sort of the same traits. They are going to cross over, but the genetic material there is going to be for the same trait. So for instance, hair color. So one chromosome may have a copy for, let's say, black hair color, and one might have for brown, and then they're going to cross over, and then the black's going to be here and the brown's going to be here, if that makes sense. So it's crossing over and they're exchanging that genetic material for that specific trait. So then the chromosomes are pulled apart in anaphase 1. So they're lining up, and then we pull them apart in anaphase 1. Then we see telophase 1 and cytokinesis. So now the cell is pinching them apart in the middle, and we end up with our 2 daughter cells. So cytokinesis technically isn't we're not look well, it's sort of taught differently, I guess, but we're not looking specifically at cytokinesis, but you need to know cytokinesis is when they're pulled apart, they're forming their nuclear membrane around the cells, and it's done, right? So that's when we've got our two separate cells. So that's cytokinesis. But in the and like the formula that you would have learned for it, for example, or generally you don't really consider cytokinesis. But it is happening there to then pinch the cells apart and make them their own little cells, so that it's ready then for the next part of it. Then individually we see prophase 2 in the two daughter cells. You can still see that it's crossed over now, so that occurs here. That is contributing to biodiversity when those genetic traits are being exchanged. So that's something that we'll come back to again in module 6. So we've got prophase 2 happening here, where we've got our two daughter cells. From there, we've got metaphase 2, where these chromosomes line up at the equator. Like I said earlier, the crossing over and the recombination occurs here in prophase and metaphase. It's not happening again in meiosis 2. It's already happened here. Now we've got the two daughter cells, metaphase 2 again. They're lining up. They line up at that equator, and we've got the mitotic spindles that are holding on to them. In anaphase 2, the sister chromatids are pulled apart. So in telophase 2, what ends up is now the cell pinches them apart, and finally, after cytokinesis is done, we end up with 4 granddaughter cells over here. You can see that they're all different. They're not going to be similar. Again, that's biodiversity. So you've got 4 daughter cells ready to go, 4 haplodaughter cells ready to go. So they're going to have 23 chromosomes. Now, what is the role in the, well, actually, before we go into the role in the body, let's specifically now look at cell replication when we see the DNA being copied here in interphase, right? So what's happening here? So let's start off with our DNA structure. So we've got the nucleotides, that's phosphate plus the deoxyribose sugar over here. So this is one nucleotide. So you see the sugar, the phosphate, and then we've got the nitrogenous base, which is a C here, cytosine. It's double-stranded, right? So it's got a double-stranded helical structure, alpha helix, and complementary base pairing. So that's Chargaff's rule, which means that the number of A nucleotides, so the number of adenosine, will equal the number of thymine, and the number of guanine will equal the number of cytosine. So they're going to be equal, they're going to be complementary, and that's called Chargaff's rule, but that's just what it is. It's going to be equal. So we've got a sugar phosphate phosphate backbone, and we've got a nucleotides here, and it's in a helical structure. It's being wrapped up. Why? Because it's long, the DNA is long, so it's wrapped up in those chromosomes, and that's where our genetic information lies. So how is DNA replication occurring? I'm going to give you a rundown of it. So we start off with initiation, where the DNA is unzipped by helicase. It's unzipped by the enzyme helicase. Remember, when we've got DNA replication occurring, enzymes play a very important role in it. So you'll come across a range of different enzymes. And we start off with helicase, which is one of the big ones because it's unzipping the DNA. Then we've got elongation, so primers bind to the end of the DNA strand. So we've got a replication fork here, so that's where it's starting. Remember, there's a 3' end and a 5' end as well. And we've got a replication that starts from the 5' end. So it's going straight down, but from the 3' end, the replication is going to start over here at the end and go up. So over here, replication starts at the beginning. Hopefully you're able to see my, is there a way to... Let me see if I can... No. Okay, I was trying to get that laser thingy, but I don't know, I've only done that on Google Slides so far. So, but hopefully you're able to see my mouse here. So with 5' end, you see DNA replication happening here from the start. But with the 3' end, this is the 3' end, the replication starts here, kind of at the replication fork, and it's moving backwards. So we've got DNA polymerase over here. So primers bind to the end of the DNA strands. DNA polymerase binds at the primer site. And from now, DNA polymerase is reading the strand and attaching that complementary free-floating nucleotide onto a nucleotide in order to get the complementary DNA. So termination, Termination is the last process where the polymerase reaches the end of the molecule and it falls off. So we've got a DNA polymerase, it's done its job, it reaches the end of the molecule and it falls off. The strands recoil into a double helix. Why? Because the DNA is long. Think of it as, a surface area thing. It's wrapped up, right? So it's wrapped up, it's coiled, it's tightly coiled in order to be small. And then we see proofreading occur by the nuclease enzymes. So it's a crash course of this little, this huge process. It's a little crash course, but what it's telling you basically is from here on the chromosome. Now remember, when DNA replication occurs, when it occurs specifically when we later on look at, like protein synthesis and stuff, not the whole thing is being replicated right now. For cell replication, it is, but later on, it won't be the whole thing that's being replicated. It's only part of the DNA that we need. So don't worry about that just yet. I'll talk about it again. But what I'm trying to tell you is it opens up, it unwraps, and we are replicating a specific part. And remember, there's a 5' strand and a 3' strand. The 5' strand is called the leading strand. And so DNA polymerase is going to start attaching nucleotides from here, from the beginning. But with the lagging strand, the 3' strand, it's happening here from the back, where it starts off at the back and it's going forward, and here they're just happening in opposite directions. That's what you need to know. Okay, like I said, it's a lot going on there. I know it was a lot, so if you've got any questions about anything you want me to go through again, please pop it up on the slide. I've been logged out here again, so I'm just quickly going to log back in and see how we went with that. If there are any questions, please pop them up, and we're going to come back to these questions. Okay, let me just grab a quick drink. Okay. Let's have a look. How we're all going here on Slido, what kind of questions I've got so far about the process that maybe I can quickly answer before we go ahead. Okay, so I've got general questions so far. Okay, so nothing specific to the content just yet, but if you've got any specific questions about anything that I've gone through right now, please ask. All right, I know it's a lot, so don't hesitate to clarify anything that I've spoken about so far. Like I said, it's a lot. All right, so feel free to ask questions, but I've got general questions so far, which is good. So now let's look at DNA and polypeptide synthesis. We know that this cell replication is happening. It's a huge process. DNA is being replicated. We looked at meiosis. We've got our 4 daughter cells ready to go. But now, why is it important, and how does it impact polypeptide synthesis? So polypeptide synthesis, you're making protein. So we looked at DNA replication. Now we are looking at transcription and translation, and how this whole process is going to give us proteins over here. Okay, so transcription. Now, in transcription, we're using mRNA. Now we are moving from DNA to RNA. What is the difference between the two? In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. So when we've got our backbone over here, where is it? Where's the backbone? No. Just missed it. Okay, here we go. So that backbone over here in DNA, it's made up of deoxyribose sugar. In RNA, it's made up of ribose sugar. And what happens is we chuck the T out for an U. So that's what's happening here. So we are replacing thymine with uracil. Again, you don't need to know why that happens. It does happen. There's there's chemistry behind it that's to do with C and G, but you don't have to worry about it. All you need to know is that we've got the U there. We've got uracil coming in. So we've got uracil coming in. So we see UGGCA. So uracil replaces thymine when we get to RNA. So transcription. RNA polymerase binds to the promoter. Now remember here, Now that we are making proteins, not the whole DNA is coding for proteins. Only a specific part of it is coding for proteins. So what that means is we're not going to open up the whole DNA. That's going to be time consuming. It's huge. We don't need the whole genetic material. We only need the part that we need to, that codes for the protein that we're after. So we've got the RNA polymerase now starting off. You would remember that DNA polymerase played a huge role in replication. You know, it was the thing that was adding on the nucleotides. Now it's going to RNA polymerase that binds to a promoter sequence, the sequence that we're using. So RNA polymerase moves along. So I'm seeing questions come through. Please keep them coming through. And okay, so RNA polymerase moves along the DNA strand. It's unbinding that small region of the DNA that we need for for protein synthesis, for polypeptide synthesis. Same thing. RNA polymerase reads the DNA template strand, so it's reading this template strand over here, and it's matching the free floating nucleotides to create the mRNA. It's an mRNA DNA strand. mRNA strand over here. It's an mRNA chain. So remember, key thing happening over here in the chain is that thymine is replaced by uracil. So we see the free floating nucleotides complement. They form whatever part of the DNA we need. So we've got that. And then the terminator sequence ends that transcription. Whatever part we need, we've done it. And the DNA is unwound over here, as you can see. And then it's rewound over here. It's whatever we need here, the template strand. So terminator sequence, then we've got the DNA. We've got the DNA as RNA that we need. That's done. And we're going to terminate the sequence. We're going to end the transcription of the DNA, and then the newly formed mRNA molecule, it's released. Where is it released? Before I go into that, there are splicing and post-translational modifications that are occurring, and we kind of go into this a bit more when we look at mutations in module 6. So I won't jump ahead right now. So we see that the RNA strand is created. It's mRNA. Like I said, not everything here is important. Introns, they don't code for proteins, so we don't need that. So we don't need them. We need the exons because they are coding. They're coding regions of the DNA. So splicing occurs, and then we've got mature mRNA. This is the mRNA that we need to then go on for translation to occur to form a proteins. So how does that happen now? That's translation. So remember, transcription, translation. Those are the two processes of polypeptide synthesis. Replication is occurring, and then we're using that to go ahead and form a protein. So translation, the mRNA docks to your ribosome. This process, translation, is happening in the ribosome. So the ribosome matches a complementary tRNA molecule, now it's tRNA, to the mRNA by matching the codon and the anticodon sequence. So we've got the growing, so as you can see, peptide synthesis is happening. We've got the messenger RNA over here, U, G, G, A, G, A, U, U, C. So that to us right now is pointless, right? It becomes useful once we get to our amino acids. So then we see tRNA over here matching it with the complementary codons. So G, U, U, A, right? And then that sequence over here, CUA, is forming this amino acid, ASP. So it's got a proper name for it that I can't remember right now, but that's the short form of it. You would, if you've done module 5, you would see that table that you work with in order to look at the anticodons and being able to make those amino acids. So we see that happening over here. So we've got incoming tRNA that's bound to the amino acid, and that growing peptide change. That growing peptide change is happening over here. So we've got our amino acids over here. Now what happens is that, so let me go back to here. So mRNA docks to ribosome. The ribosome matches a complementary tRNA molecule to the mRNA by matching the codons. These are codons to the anticodons over here. As subsequent tRNA molecules dock, a polypeptide bond is formed. This is a polypeptide bond. The polypeptide bond is formed between adjacent amino acids. The polypeptide The polypeptide chain is elongated by continued addition of those amino acids. And when a stop sequence is reached, there is a stop sequence. Again, if you've in that table that you would have looked at, should have put an image up here. I'll remember that next time. But you've got that table that tells us what the stop codons are as well. So we've got that stop codon, it's happening, and then it gets just stopped. Remember, because we don't need the whole thing. It's mRNA is just part of the DNA that we need, right? It's just giving us that part of the DNA that has the codes for protein. That's all we need. So there's a stop and a start codon. It doesn't keep going. And then it stops, right? So when the stop sequence is reached, the ribosome releases that mRNA and the polypeptide molecule. We're done. We're letting go of the mRNA molecule and that polypeptide molecule. The polypeptide folds and it undergoes, again, this post-translational modification that results in a mature protein that's ready for use. Why does it go through that post-translational modifications? Because remember, it's making sure that, you know, we've got what we need. And we'll come back to this when we look at mutations happening in module 6. You know, if there's, for instance, a wrong code there, whatever, we need to make sure that we've got the right code, and that's where those post-translational modifications are happening. So now we know that the protein is formed, we've got polypeptide synthesis done. So now we've got our protein structure and function. What is happening here? So our fundamental unit is amino acid, right? So that's a fundamental unit of protein. So amino acids. Sorry, if you're doing chemistry, you would be familiar with amino acids. You know, you'll look at the formation of amino and carboxylic chains as well. And of course, you know, look at all of these different chemical molecules and how they play a role in DNA synthesis. But right now, we don't need to, you know the chemical structure. What you need to know is what that unit is, what is it doing. So functional properties of amino acids and proteins determined by the side chain chemistry. So there's, you know, obviously polar, non-polar, negative charge, and positive charge. So we need to know what's happening over here in terms of, you know, polarity, and polarity, what it's going to ensure is, you know, where the molecules are pulled. But in your head, just know that amino acids are functional properties of, lost track of where I was reading. Functional properties of amino acids and proteins, they are basically determined by the side chain and the charge of the side chain. Then we've got a primary structure, so the sequence of amino acids in a polypeptide chain. So that's our primary structure, just the amino acids, and however they're structured in a polypeptide chain. The secondary structure, with this, you know, it's not. I'm pretty sure it's not listed specifically in the syllabus, but what it does is it's just about knowing those fundamental units and, you know, having that, being able to know what is it doing. So we've got the primary structure, then we've got the secondary structure, the formation of alpha helices and beta sheets. So that's all dependent on the hydrophobic interaction. Hydrophobic, remember, is water hating. Hydrophilic is water loving. So not all parts of the amino acids are going to be hydrophobic, but the parts that don't like going water, they are the ones that are going to be positioned in a way that they don't interact with, like, the water in your cells and stuff. So that's basically going to lead to the formation of alpha helices and beta sheets. That's what you need to know. Again, we're not going into, like, super depth here. It's just knowing what the basic processes. Then the tertiary structure is that formation of the overall 3D structure, the side chain interactions, you know, how the side groups interactions twist the backbone for maximum stability. So this is when we're getting to that 3D structure of the structure. And finally, the quaternary structure. So 3D structure of the protein is the tertiary structure, and then the quaternary structure is when multiple protein subunits are interacting with one another. I know I said a lot there. I'll break it down again. So we've got primary structure, that's the amino acids on a polypeptide chain, that's your basic amino acids. Secondary structure is when when we basically see the interaction of those amino acid molecules. You know, they are hiding away, parts of them are hiding away from the water, they don't like it, so they're being structured in a way that forms alpha helices and beta sheets. Then we've got the tertiary structure, that's the whole 3D structure of the protein. And finally, quaternary structure is when multiple protein subunits are interacting together. So what are our functions here? We've got support and structure, so macromolecular keratin and micromolecular tubulin. So macromolecular big molecules, molecular small molecules with macromolecular keratin used in our hair, in nails. That's what, that's in our nails too. So it's used for bodily functions. Then we've got enzymes. So you would see, for instance, we've come across some enzymes here when we looked at replication. So those, and there are lot more in your body. So so enzymes, for example, they're biological catalysts, and they are made from protein. So for instance, ATP synthase, they are protein. Antibodies, so essential part of the immune systems, for example, immunoglobulins. That's where protein is coming into use again. Then we've got hormonal messengers like oxytocin, and finally transport and storage, where protein, for instance, moves molecules around the cells and around the body. For instance, moving around the ferritin stores in the body, the iron stores. So basically, you need to know that protein is an essential unit in the body. It's important. That's the structure, and the functions are that it provides support and structure. It functions as enzymes. So enzymes are made of proteins. Not all though, but like most of them. Antibodies, messengers, and transport and storage. Again, let's now move on to genetic variation. So we're getting close to wrapping up this module. We've got one more to go. I have been signed out again on Slido here, so I'm quickly going to jump back on and see if there are any specific questions to what I've just gone through. Like I said, there's a lot going on here, lots of different concepts. If you have any questions, please ask. If there's anything you would like me to clarify, please pop it up, and I'm happy to go through it again. It's no problems at all. So just waiting for that to open up. Let's have a look. Do we have any burning questions? Okay, we've got lots of questions up here. Okay, so I've got two here that I'm just quickly going to go through. Are there any differences in RNA, any other differences in DNA and RNA? Well, the main, basically the two that obviously the main two that we need to know about is that, DNA, there's that, and those are the main two, is that, we've got deoxyribose versus ribose sugar, and also that the thymine is replaced for uracil. Okay, there is of course molecular stuff going on, but you don't need to get into that. You just need to know these main two. You can obviously go ahead and research, for instance, how the thymine turns into uracil. It's quite an interesting process. I won't go through it, I don't want to confuse you, but you can go ahead and research that if that's interesting. But those are the main two that you need to know about. What is the promoter sequence? So remember when we looked at DNA, so I've got two questions. What is the terminator and promoter sequence? Let me just go back here, pull up that diagram. Okay, so promoter sequence, that's what we're looking here. Okay, so remember, so we've got our terminate, where is it? Can't find the image. Yeah, so remember, within the promoter sequence and the terminator sequence, that's the part of the DNA that we need. So we've got the promoter sequence that we start off with and the terminator sequence that we're ending off with. We also see, and yeah, so that's the main one over here. Yes. Okay, and also when we come to our transcription over here, the terminator sequence ends the transcription for the DNA. So that's the last sequence over here that's ending the transcription. That's where we stop, right? And the promoter region, the promoter sequence, is where we're starting. So this is where we start. Remember, in so let me rephrase that. In cell replication, we're looking at the DNA being replicated, all of it, but within transcription, we're not replicating the whole thing. We just need the part. The promoter sequence is where it starts and the terminator sequence is where it ends. Just that part from the promoter sequence to the terminator sequence, where we've got the coding region of the DNA that's going to code for proteins. That's all we need. So that's the promoter sequence where it starts off, and terminator sequence where it ends up. Sorry about the confusion over here. I got confused here for a second with the diagrams. But yes, we've got, so just to repeat myself over here, we've got the promoter sequence in transcription where we start off that coding. So remember, we're not coding for the whole thing. We just want this bit over here that's going to code for my proteins. That's all I need. So I'm going to start off over here, my promoter sequence, and end up over here at my terminator sequence. That's going to end the transcription of the DNA and give me my fully formed mRNA that I need for that specific part of the DNA there. I hope that helps. If you've got any more questions related to that, please let me know. All right, but hopefully that helps. Okay, moving on. Okay, yeah, maybe I should have just gone the other way, but that's all right. So we are now looking at genetic variation. So how can the genetic similarities and differences between and within species be compared? So we've looked at replication, we've looked at all of transcription, translation to know what's happening at a molecular level. Now we are zooming out and looking at how do they differ between species, what's happening over there. All right, so let's have a look. So let's firstly talk about the chromosomes. We've got these are autosomal chromosomes. There's 22 of them, and then we've got sex-linked chromosomes. So 2 of them over here. So remember, altogether in our body we've got 46 chromosomes. So yeah, so 22 these bad boys, and then 2 little babies here. Those are sex-linked because they are determining the sex of the organism, right? So you've got X or Y chromosomes, and then these are autosomal ones. Remember, you've got 2 copies over here, so one from mum, one from dad. So altogether they're going to be 46 chromosomes. So 22 pairs, but altogether it's 46, including these over here. These only here for X, you'll have if it's X chromosome, you'll have 2 of these strands. This here is a Y chromosome. So you can tell by that one big one and that small one. So that's X, that's Y it says here. If there were 2 big ones, that would be X. So XX is female, XY is male. Yeah, so that's that without confusing you. So I'm just making sure we're going through these over here. Now, looking at the different traits, how are they expressed? So we've got dominant, so trait will always be expressed over other traits. And recessive, it requires 2 of the same allele to be inherited in order to be expressed. So the way that these are interacting with each other is going to impact how, what specific traits you have. And that's basically your phenotypic expression. So for instance, I've got black hair, so that's going to be based on how these different chromosomes, like not all of them are coding for hair, of course, but and I don't know which specific one it is, but let's say how number 12 is coding for that. And if both my parents have black hair, so I may have that. I may have a recessive allele. I've got black hair, parents have black hair. So that's what it is. So what I'm trying to say here is how these are interacting with each other. These chromosomes are going to impact the phenotypic expression, how you look. So if it's a dominant trait now, it will be expressed over all other traits. So if in my genes, black hair is the dominant trait, I'm going to have it. It's going to be expressed over other traits. So I may have had a recessive, light brown hair trait, but if black is dominant, that's how it's going to be expressed over that light brown one. Then recessive, so it requires 2 of the same allele to be inherited in order to be expressed. So recessive alleles, this is when we now look at Punnett squares. And I saw one comment here, so I've saw a couple of comments here in terms of, if it's a bit confusing and you haven't done mod 5 right now, don't worry about it. I totally get it, right? So what I just want you to focus on is maybe capturing some of this terminology. What's autosomal, what's sex-linked, what's translation, what's transcription. Just think about the processes. Won't make sense immediately. Even when you start module 5 in class, it takes time to wrap your head around it. Definitely took me time, lots of questions to wrap my head around, okay, where is this process occurring? How is it occurring? What is it? It's confusing, right? So just think about the big processes that we've talked about today, and maybe try to research them a bit and try to just understand them. And hopefully when you once studied in class, it would make a lot more sense. As I also said, it's a revision for module 5, so I'm not going into a lot of detail. I'm kind of just going through the major concepts. But if you've got any specific questions, please ask. So traits will always be expressed over other traits. That's dominant, and so the name itself will help you remember that it's a dominant trait. It's going to be expressed over others. Recessive trait, so if both, let's say, both my grandparents had light brown hair, right? And then that trait, it's not It's not phenotypic, like my parents have it in their DNA, but it's not expressed. They've got black hair. But then in the third generation, which is me, I might show it because it's in both of their DNA, and then I might show it's recessive. So it's when you've got two of those, it may not be in every generation, it may skip a generation. Something like that. Think, for instance, about red hair, right? So like got a family friend, they both both of my uncle and aunt, they both have black hair, but their daughter has red hair. And it was, their grandparents, someone had red hair. So it's not dominant, it's not happening in every generation, but the parents would have the recessive alleles for it, and that's where it's popping up in the offspring. So I know it's a lot. I totally get it. It's just trying to think of the concept. Dominant, it's the trait will always be expressed over others. Recessive, you need two of the same. So so in order for it to be expressed, it needs to be there. Your parents may not have it. For instance, that specific family friend that I was talking about, both the man and the woman, they didn't have red hair, but their ancestors had it, great grandparents and stuff, so then it showed up in the baby, so she has red hair. Okay, so codominant, both alleles in a gene pair are fully expressed. So we've got both alleles here. So we have alleles here. What are alleles? So let me go back here. Okay, so we've got both alleles over here. That are being expressed. So I've got allele for black hair and allele for black hair on both of these okay, on both of these chromosomes, it's being expressed. All right, so an allele is basically that trait, that's there in the um in the genetic material, in the DNA over here. So I've got an allele for black hair and an allele for black hair over here, if that makes sense. All right, I've been logged out of Slido again, which means I can't see your incoming questions, but let's not worry about that for right now. I'm going to spend a lot of time on questions, so bear with me as I go through these. Think of these big ideas, jot them down, and then maybe do some of your own research. That can have a huge impact on your understanding. So incomplete dominance is when an allele is not completely expressed over its paired alleles. So if I've got, let's say in flowers, if there is a pink flower, a red flower, and a white flower, they are mating. So they're mating. So we've got pollination occurs and everything. So we've got a red flower and a white flower. Incomplete dominance is when, let's say the red red allele is dominant over the white allele. But incomplete dominance would be when the red allele doesn't win over the white, so you will end up with a pink flower. That's what it is. That's incomplete dominance, right? If they were codominant, red and white, I might see, I'll have a flower that's a mix of red and white. Not pink, but you see red and white specifically. If dominant would be, the red would win. I would have a red flower. Recessive would be, the red will not win. Even though it's dominant, it won't win. The white will win. It's recessive because the white is recessive. And then multiple alleles is when an allele is not completely expressed over its paired alleles. So as you can see over here, if you've got a brown and white rabbit, so you may see a variation of these colors. So that's what it is. Again, if you've got any questions, please pop them up. I know it's a lot, so just try and think of these big concepts and then do some of your own research. Play the slides, play the recording again, so that you are able to maybe slow me down and then go through it again. So now we look at pedigrees and Punnett squares. These are so helpful when you think about it. There's a lot of concepts going on. It's quite mind-boggling if you are not able to visualize this somehow. So pedigrees, that's about tracing phenotypes through generations. Remember, phenotypic traits are the traits that you can see. So if you've got black hair, that's your phenotypic expression. If you've got brown eyes, that's your phenotypic expression. It's whatever you can see. So they help us trace phenotypic expressions. So if we look back to that, that hair color example, again, grandparents may not have had red hair, great grandparents may have. So then I see it as a recessive allele that pops up in the great grandchild. So it helps us trace phenotypes through different generations. You always need to include a key, and if you see a pedigree, like one that's given in the exam, for example, it will include a key. it will tell you, but it's just certain, like the circle represents female, square represents male. The affected trait, the person affected with the trait will be colored. The diseased, there's that cross there. Twins, you'll see them this way, adopted, and a miscarriage. So this is the generation number. So the first generation may be, for instance, parents. The second generation is children. That's the line of the descendant, and that's a sibling line. In twins, it would be like that. So, recessive traits skip generations, dominant cannot. So, like we spoke about earlier, recessive traits will skip generation. In the hair color example, great-grandparents had red-colored hair, grandparents didn't, parents didn't, but the child might have it. It's a recessive allele that has skipped 2 generations. That's what it is. If it was a dominant thing, if red hair color was a dominant allele in their DNA, then it would pop up in every single generation. It will not skip generations. Sex-linked traits affect one sex or more, right? So it may affect both the daughter and the son. The best thing with these is to practice these questions a lot. You'll find a range of these questions, obviously in the biology papers. You'll find them online. You can find them in the biology sample paper that's published by NASA. So doing them will make you feel more comfortable with them. Now, Punnett squares, they allow us to theorize how many genes pass from parents to offsprings. So I've got an allele, right? So I've got, let's say, BB. Big B means it's dominant. Small b means it's recessive. So if I've got BB, this is so we can see here, this is homozygous. No, this is heterozygous. So you see BB, it's 2 different. It would be homozygous if it was 2 capital B's or 2 small b's. But if it's big B and a small b, that's heterozygous. It's 2 different. So we've got pollen here from one flower and pistil here. So that pollen has this trait BB, and the pistil also has the same trait. So these 2 can interact with one another, and we'll end up with a homozygous dominant trait over here. Big B, small b, we end up with a heterozygous trait. And over here, what's going to be expressed? Think about it for a second. Out of these 2, which one is going to be expressed? It's of course going to be the big B. That's going to be the trait. So if big B is purple colored, yep, so it's purple. So that's what the trait is going to be. The flower is going to have purple petals. Here, the flower is going to have purple petals because it's 2 capital B's. Here, the capital B wins over the small b, so it's going to have purple. Same thing over here. I've got a capital B and a small b. We're talking about purple color, purple petals, so it's going to be big B and small b. Purple is going to win as a phenotypic expression. Finally, I've got 2 small b's, so that is going to be the recessive allele. So it's going to be homozygous. But it's not the dominant one. So that means we've got a white flower here. This one wins because it's 2 small b's. They're the same. So I know it's a lot, but I'm just quickly going to go through one more time because I totally get it. We've got 2 capital B's over here. All right, so that's going to result in homozygous dominant. So it's going to have purple flowers. Over here, I've got the big B and the small b. Big B encodes for purple color. Small b encodes for white color. Big B over here is going to win. So we're going to end up with purple colored petal. Same thing happens over here. And then in small bb, we see the white color. So we see white color because that wins. It's homozygous. Okay, so key. You want to have a key here that actually tells what's happening, right? You'll be asked to make Punnett squares. You may be asked. Won't say you will be asked, but you may be asked to make them in the exam. So you need to have a key. There needs to be parental phenotypes and genotypes, and this was the parental phenotype. This is what was expressed in the parents. All right, they are heterozygous over here, and then over here we see the genotype. But if the thing represents whatever, okay, so remember the genotype is what you encode for, but the phenotype is what is expressed. So that's the difference between the 2. Then we've got offspring genotypes plus phenotypes. You need to be able to show that as a percentage. So 75% it's going to be homozygous over here, but 25% it's homozygous dominant. 25% it's going to be homozygous recessive. Then percentages and ratios too. So over here we see the ratio of 1 to 1 to 1. So 1 homozygous dominant, 2 heterozygous, and then 1 homozygous recessive. Okay, now let's move on to that last bit on inheritance patterns in a population. Now we zoom out even further. I'm quickly just going to open my slider again. It's locked me out again. See how we're going. That was a bit confusing in terms of the Punnett squares and pedigrees, what's happening there. But just, again, think about these concepts, watch a couple of YouTube videos. They always help. Like, they always help. Personally, I loved watching them. I found Amoeba Sisters quite helpful. And there's a lot more like Khan Academy, Crash Course, all of them. So have a look around, see if you find some of those helpful. Research those concepts and try and make sense of them in your own words. That can be super helpful. And of course, ask questions. I'm just popping up here on Slider again. See what kind of questions I've got in terms of the content. There was a lot going on here. Okay, it's not logging me on for some reasons. Give me 1 second. Okay, still not working. Alright, what I might just do is, if it doesn't work this one more time, I'm going to leave that and then have a look at the questions once we go through the poll. Okay. Give me 1 second. So yes, right now, if you've got any questions specifically, please ask. I'm on it now. I'm going to answer it as we go through it, so that way I'm not coming back to it later. If it's a content specific question, please ask now about any of the things that we've gone through currently. Okay, still working. Alright. Technology has its days. Okay, I think it's working. Okay, it's working now. Give me 1 second. If you've got, okay, I've got lots of questions. Okay, yep, so I've got lots of questions. Okay, let me have a look. So, okay, what I might do is I've got them here. I'm gonna send them through for now, and then because we've got a break after this, so I'm gonna come back to them, and have a look, and then go through them. All right. So because there's a couple, so I don't want to sort of stop in the middle and then go through them, but I'll come back to them and the good questions. All right. So keep asking. Like I said, I know there's a lot happening here. I'm trying my best to break it down, but if there's anything that doesn't make sense, any of the examples, feel free to ask again. So inheritance patterns in a population. This is that last little bit of this module. It definitely, by the time you get here, you can definitely feel like it's been a long, tiresome module. So have some patience here. It's not a big dot point. It's just looking now at the big picture of how can population genetic patterns be predicted with any accuracy. Why is that important? We'll talk about like preservation and conservation strategies, and we'll talk about why that's important as well. Okay, so DNA sequencing. Let's start off with there. Firstly, how are we going to predict them? How are we predicting DNA patterns? Now, why did we look earlier at pedigrees and Punnett squares? So that we're able to now, look at and predict the different variations of like phenotypic expression and stuff. Now we look at how we can actually DNA sequencing, how I can go ahead then and look specifically at the DNA where the DNA is coding for that specific allele. Remember in the chromosomes, at some point, there's going to be a little allele that codes for my hair color, or my eye color or something. So I may specifically want to look at that and, extract that or something. So we're looking firstly at DNA sequencing. So firstly, isolating DNA from the cells. So if you're looking at DNA sequencing, we need to isolate DNA from the cells. Remember, DNA is going to be in all of your cells, so we're isolating that. Secondly, identification of sequential order of nucleotides. We are going to need the specific nucleotides, that specific part that is going to perhaps code for a certain trait or code for a certain, and this is helpful, even when we look at, for in crime shows and stuff, you would have seen the investigator comes in and they're taking part of, your hair or something that's found on the crime scene. They're going in, putting into the lab. finding the DNA and using that in order to identify the person. We kind of thinking along that track here now. So we identify identification of sequential order of nucleotides. There is computational processing here. So now we are comparison of whole genomes. I've got someone's whole DNA. I'm comparing it. I'm comparing the stain found, let's say, on a knife, to the hair that's found on the strand of hair that's found on this crime scene, and comparing whether those DNAs match or not. Right? I think when you think about it in terms of mainstream media and the depiction of it, kind of makes it a bit easier. It may not be used for that specifically. There's lots of other uses that we'll talk about later too. But that's like a really famous one that you would have come across. Okay, so we've got computational processing occurs, we are comparing the whole genomes, then from there, transcription and translation of genes inside here, so that's a specific program here. Then single nucleotide information. I'm finding that specific information over here. Each individual has different genome. Remember, that's what makes us unique, having different DNA. Identification of differences and similarities. So whatever the handprints that are found on the crime scene versus the hair strand that I found. Now I'm looking at whether those are matching. What are the differences and similarities? The whole thing may not match, right? Due to chance, like the whole thing, well, more than likely the whole thing will match if the person is the same, but sometimes it can be two different people, but parts of the DNA match. We do see that that's something that's popped up, as a proper thing in the specific field of area where there is a unreliability in this process. But for now, just think about that identification of differences and similarities. Modern computation allows that pattern to be identified, and then we trace inheritance of genes, alleles, and SNPs between people. Now, from there, DNA sequencing, I've got a DNA, I'm able to sort of see the differences between the DNA. Now I look at DNA profiling. I collect DNA samples from cells, common practice, blood, hair follicles, mouth swabs, digest the DNA. So we cut the DNA into small pieces, and we use that restriction enzyme to sort of just see, what part of the DNA are we looking at, what allele are we looking at. We look at hair color or eye color or something. Or so remember, we're just looking at, we can look at the whole thing, but we more than likely are using a restriction enzyme to look in specific places. Then we've got DNA fragments are separated by gel electrophoresis. So it's a machine, basically. You put the DNA in it, and then it's using electricity to kind of the DNA is moving. So over here, it's a bit hard to explain without having the actual machine there. Or if you haven't done it, have a look at YouTube videos of gel electrophoresis. That will definitely help, where they've got actual machine and they show you what's happening. But to explain it to you, I've got the DNA over here. Let's see, I've got samples A and C. So A is what I found on the crime scene. C is one of the suspects. I'm going to put the DNA over here and here. Then once I start the machine off, the DNA is going to move according to the electricity. It's moving forward. When it stops, if they're at the same places, actually I've got a marker here. A is where I'm putting the crime scene over here. A, B, and C are my samples. So let's say C is the suspect, I've got the marker here. The thing is moving, and then once it stops, you can see the similarities over here. So C matches over there, C matches over here as well. That's B, and then C is matching in 3 specific spots to the marker. A is only matching in 1, and B is only matching in 2 places. So is that giving me concrete evidence that it's C? No, because not the whole thing is matching. But it is telling me that C, I need to investigate C a bit more maybe, because that matches more than A and B. But this is where, again, that unreliability and uncertainty arises, because sometimes, it can happen and it may not even be that person. But anyways, focusing here on the process, collect DNA samples from the cell, digest DNA, cut the DNA into small pieces using a restriction enzyme. So I'm just putting in the small piece over here. DNA fragments are separated by gel electrophoresis. The gel visualizers show a band patterns. These are the band patterns, and that's what it's showing over here. And fingerprints can be compared. So it's specifically looking at fingerprints. Visualization of fingerprints allows for comparison of patterns. The degree of similarity equals the degree of relatedness. So basically, what we're saying over here is that A, is not as related to the marker in comparison to C. So there's that degree of relatedness. It's not, remember, it's not, it's correlation, but it's not like, so there is a correlation between them, but I can't say that it's not causation. But what I basically mean by that is that I'm saying that there's that similarity, but it does not give me like direct proof that C is a culprit. Not saying that just yet. There'll be more investigations done for that, but from a science perspective here, we're thinking that the degree of similarity in those band patterns is going to be the degree of relatedness. So we may observe conserved regions of the DNA and effective in humans due to large stretches of junk DNA. We'll be talking a bit more about junk DNA in module 6 as well, but we do see that there's that it's effective in humans because of the uniqueness of the DNA. Okay, now let's have a look at where does the whole thing come into play. So we can use this in large-scale collaborative projects. So from there, we can gain trends, patterns, and relationships in order to then apply those findings and put in place distinct projects or distinct distinct projects or distinct regulations or something, especially when we are looking at conservation, animal plant conservation. That's why it comes into a huge place. So conservation management, so conservation genetics, fields combining molecular genetics, ecology, and biodiversity sciences in order to propose species management strategies. Uses population genetics to identify alleles at risk. So if I'm looking at, and this is used, when we look, again, Slido check me out, but there were no questions so far. If there are any questions, please pop them up. But anyway, back to conservation management. So, if we look at animals that are going extinct, a big example of that is, for instance, Tasmanian devils, right? So if you're looking at that, we're looking at what specific alleles are putting them at risk and how can we counter that. And we find that, for instance, by looking at devils that have already passed away, looking at their DNA, what was there, versus an animal that's sick, seeing if there's, let's say, any similarities between that, between the DNA patterns over there. So it uses population genetics to identify alleles at risk. It proposes ways to manage and preserve biodiversity. So Australasian Wildlife Genomics Group at the University of Sydney, they have a project going on. So the Devil Tools and Tech project, they've got the Koala Genome Project and Marsupial Genomics. So they are working closely with the Australian Museum, Chicago Zoological Society, and National Cancer Institutions, for instance, in order to use population genetics to identify what alleles are at risk and then counter that by proposing ways of managing biodiversity. So yeah, that's that. Now, inheritance of disease. So only a 0.8 nucleotide variance among humans. So all genetic disease must be contained within this variance. That's the variance that we have among humans. So any genetic disease that we've got, they are contained within that variance. So understand the cause of genetic diseases. For instance, is it hereditary? Is it hereditary due to random mutation or by what interaction of the alleles? We are then able to design therapies to repair the genetic disorders. So Human Genome Project. So collated data on thousands of genomes to identify genes, variable regions, haplotypes, etc. The Human Genome Project was quite big in the 1990s and early 2000s because it was a big scale collaboration within scientists around the world where they looked at mapping the human genome. So it was huge, it was new technology, a big deal. The US government invested a lot in it, and so did other institutions as well. So collated data on thousands of genomes to identify genes, variable regions, and haplotypes. They have identified genetic causes of breast cancer, Alzheimer's, and colon cancer as well. So this basically, when we look at being able to have the genome of thousands of peoples, hundreds and thousands of peoples, will then allow them to basically go ahead and look at, you know, what are the similarities and differences in the interaction of alleles within these distinct genomes. If we look at hereditary diseases, or we look at and we look at hereditary diseases, like for instance breast cancer, where are we finding the interaction of the alleles that tells us, okay, they are at risk of breast cancer, this is what the risk is, and then how can we counter that? So it really does tie in with the bigger sort of scientific question of, you know, improving healthcare, and having better resources for people. So then we've got human evolution. So within that, we are looking at cultural and regional groups, and often linked by prevalence of particular haplotypes. Mapping haplotypes allows us to trace the movements and evolution of human species, and identification of common ancestors too. So we will talk about mitochondrial Eve, and, you know, how we've been able to trace back to mitochondrial Eve, who lived sorry, give me 1 second. I've got my charger playing up. Okay. Alright, what I might just do is, just got an issue here happening over here with my charger. Give me 1 second. I'm gonna have to plug in my charger. Alright, so what I'm gonna do is just give me 1 second while I do that. But I'm just gonna quickly finish off with this, and then we're gonna pop over to Slido to answer a couple of questions. What battery do I have? I've got 19%. It's gonna be enough for this. It's running out a bit faster than I imagined. I just charged it this morning, so computer problems again. Alright, but anyway, so we've got human evolution, and like I said, we have been able to trace back to mitochondrial Eve, who lived in Africa 220,000 years ago. And we've got the International HapMap project, so it uses genome data to identify SNPs, haplotypes, and haplogroups. And from this information, we are able to track lineages between populations. Alright, so my top tips for Module 5. What are they? So know your basic processes like meiosis, mitosis, DNA replication, polypeptide synthesis, and protein folding. Know your basic skills, like Punnett squares, pedigrees, right? Think about the broader context of why is it happening. When, like I said, we looked at those specific molecular processes, and then we zoomed out to look at the genetic variation. So how do we achieve this variation? And memorize your examples like methods of replication, functional proteins, methods of... Okay, so we look at Module 6, mutations. How does mutation introduce new alleles into a population? That's the first thing we are looking at. We're looking at how mutagens operate, the different types of mutations, somatic versus germline mutation, effects of mutations on coding versus non-coding DNA, causes of variation, and also population genetics. That's where we kind of look at replication again, and, you know, why do we look at those post-translation modifications in DNA? What happens, over here, when we look at effect of mutations, what happens if they go wrong? So, how does mutation introduce new alleles into a population? So, mutation is a permanent alteration to the nucleotide sequence of an organism's genome. So, how does that happen? Firstly, electromagnetic radiation. Everything around us has electromagnetic radiation, and that is a culprit. So those are waves that are emitted by electrically charged particles. They interact and ionize atoms that they encounter. Danger of radiation lies in its penetrative ability. So you would look at alpha, beta, and gamma particles, and it can travel into the nucleus and interfere with the DNA molecules. So how does that then cause mutation? Well, input of energy may alter the way that DNA is bonded, changing its chemical composition, right? So I won't go into the specific process. What generally is happening is that energy is going to shift the way, you know, the nucleotides might interact. So you see nucleotides being replaced with one another. So we'll talk about that in a bit more time. But what you sort of need to know here is that the energy is what's going to alter the way that the DNA is bonded, you know, the backbone and the nucleotides. It may cause rearrangement of the structure. So we'll talk about how that happens and what types of rearrangement it can cause. So a huge example is UV light. So radiation from the sun encountered daily. It's harmful in high doses. Incoming protons excite adjacent pyrimidines. So those are the T or C, so thymine or cytosine, causes them to form covalent bonds. So what happens here is, instead of being bonded over here, you can see that the energy is now making them bond to each other this way. That's problematic. And on an average summer's day in Sydney, a person will acquire about 20,000 pyrimidine dimers. So that's why, and that links back to skin cancer too. You get UV rays, it alters the DNA over here, and then you may see that those the cell keeps replying, like it's made alterations to the DNA, and when the cell keeps replicating itself, it can then go ahead and become a tumor. So that's where it's also, can cause cancer too. So that's why it's harmful. Huge campaign, slip, slop, slap, things like that. So that's why electromagnetic radiation can have a huge impact on the DNA. Chemicals, so radioactive agents. Uranium is a big one, that's, and you would have seen, like, for instance, Marie Curie, Rosalind Franklin. You would have looked at how huge scientists and how they were exposed to chemical mutagens, and how that affected, obviously, exposed for a long time. We are exposed to them too, but I mean, if you think about it, if you're like a normal person, not as much, but if you're working in a scientific field, if you're working in a factory or something, you have to wear protective equipment, protective clothes that are protecting you from the radiation. You would see people working in like nuclear factories and stuff, at like nuclear sites. They have to wear protective clothing, because radioactive agents like uranium, they can be very harmful to us, to our body. So radioactive agents, they release radiation, including alpha and beta particles, it's a high energy particles, and gamma waves, which is also a form of electromagnetic radiation. They penetrate the cell and interact with the DNA bonds, creating structural disruptions to DNA, as we so like we looked at earlier, they're changing the structure of these nucleotides within the DNA. Metals, also huge: arsenic, nickel, and cobalt. They affect processes of DNA repair, so they are so they can stop proteins from being able to recognize base pair mismatching. So if you've got, let's say, a C bonded to a T, they may not be able to pick up on that, right? So that reduces the fidelity of the DNA replication system, so that there are more errors that are occurring. Nickel, it inhibits ability for histones to condense DNA, so bad for DNA chromosome formation. Naturally occurring mutagens. So we see, obviously, chemical mutagens and natural mutagens, but now naturally occurring mutagens, the big example of viruses, right? Replicate by inserting their own genetic material into the host cells. If you've done module 7, you would have looked specifically, how that is happening, how they're interrupting the host cells and using the host cells in order to become stronger. They may disrupt our normal cell function and may lead to lasting changes to an organism's genome. Bacterial infections are another one. They induce inflammation, they reduce the efficiency of the DNA repair system that we were talking about, increasing the rate of mutations. And an example is the Helicobacter pylori, which has been linked to the development of stomach cancer. So viruses and bacterial infections, natural mutagens that can have a huge impact on the structure of the DNA, and also the DNA's inherent repairing system, sorry. So mutagens, now we look at what are the causes, processes, and effects of the different types of mutations, including but not limited to point mutation. So the causes, as we discussed: external mutagens, DNA replication errors. Sometimes, obviously, there are processes there are processes within the system that are designed in order to repair the DNA if there is any problems with it, if there has been any mismatched pairs or any other issues. So sometimes, and we looked at earlier how mutagens can mess with that. DNA repair system malfunction, that can happen too, and that again will affect the system. So the effect. So have a look at the table, have a look at the mind map over here, the chart over here, where we look at the effects. So we've got point mutations. This is our normal codon sequence. We've got A, G, C, AAG, GCU. So now, a quick question. I want you to think of this. is this DNA or RNA? I'm going to give you 1 second. Think to yourself, what is it going to be? And if you answered RNA, you're correct, because these are anticodons, and we've got U there, uracil, that tells us that it's RNA. But so we've got our normal codon sequence over here. So there's substitution occurring or frame shift. So substitution is when that G is substituted for a U. That's a substitution mutation. A frame shift mutation is that a mutation causing a downstream shift in the codon rating. So, for instance, what I see, what happens here is that the C is inserted here, is insertion. Because the C is inserted here, it changes the codons over here. So instead of having these three, it changes to give me the gene and gly. And then there's also deletion, where that G may be deleted itself, so it deletes a whole amino acid. Basically, it's disrupting the process. Then the classification of mutations. So, for instance, substitution. Now we look at the effect of it. So these are the classification. These are the mechanisms. There's a substitution mutation, frame shift mutation, insertion, and deletion. And these are the effects of it. So we've got missense. So missense is when the mutation which edits codon sequences to incorporate different amino acids into the protein. So the amino acids here change. Nonsense is when mutation which results in the creation of a stop codon prematurely. So, UAG over here, that's my stop codon. Because of that mutation over there, my codon stops earlier than it should, and that's obviously going to have effects. If it was a that may make my amino acid chain not finished properly, and that would impact that my protein chain is only half formed, and that's going to have an effect on the way in which that protein is then going to function. It may make the protein redundant, right? So that's nonsense. And silent is when the mutation has no effect. Somehow, I've replaced a U here, but somehow that still retains the same codons. It retains the same amino acids here. How does that happen? Look back at the charts. You can find it online. We can find the anticodon charts, and for some of the amino acids there is a variation of distinct codons. So AGU, AGC, they both code for SIR, so they both code for this specific amino acid over here. So that means it's a silent mutation. It hasn't affected the function of the protein. So like we said, the effects specifically are silent, missense, nonsense, a creation of SNPs. So it creates SNPs, creation of new alleles, potentially beneficial, and it can be potentially negative too. And just because a point mutation is small, it doesn't mean that it can't be disastrous. It can have disastrous effects, for instance, cystic fibrosis. So now, just trying to have a look if there are any questions. So yeah, so creation of SNPs, creation of new alleles, potentially beneficial, and just because the point mutation is small does not mean that it can't have harmful effects. SNPs, by the way, there was a question in the Slido 2 earlier on. So SNPs are single nucleotide polymorphisms, and they are basically the common type of genetic variation that we've got among people. So that's the differences within our DNA. So that's that. Again, if you've got any specific questions to that, please pop it up, but I haven't seen any coming through yet. So mutagens compare the causes, processes, and effects of different types of mutations. So chromosomal mutation, we look at the causes. So causes are that usually a result of error in meiosis, right? So there's error in the DNA replication. So not DNA, in meiosis, right? So it's going to affect the daughter cells. So crossing over occurs incorrectly. That can happen. So chunks of DNAs are screwed up. That's structural mutations. So we can see over here, structural chromosomal mutations, deletion. A chunk of the chromosome over here is deleted. It's shorter. Inversion. It's been inverted. So yep, it's been inverted. Translocation, as you can see, it's been changed. So the formation over here in crossing over, they've changed completely. And duplication, it's been duplicated. So it's been from here, it's popped up again over here at the top. So sister chromatids are incorrectly separated during anaphase. So when they separate during anaphase, it's happened incorrectly, which has led to these specific mutations. And that can obviously cause a number of mutations, and they can have harmful effects too. So the effects are usually severe because now it's specifically affecting the chromosomes. It's not affecting the protein, it's affecting the chromosomes and the DNA. And because chromosomal mutations involve a large number of changes to the genes. So mutation in chromosome number. So you want to kind of know these terminology here. So aneuploidy is when the overall chromosome number of the offspring is different to the parent organism. So for instance, in Down syndrome, there are 47 chromosomes instead of 46. That's because of trisomy 21, where there are 3 copies of the chromosome number 21. And polyploidy, where we've got an organism contains more than 2 sets of homologous chromosomes. So this is when, remember, we've got 2 sets of chromosomes. We've got 2 sets of chromosomes, so 22 pairs there. But in polyploidy, there may be more than 2, so there may be 3 sets of chromosomes. So triploidy is when a fetus has 3 copies of every homologous chromosomes instead of the usual 2 copies. So instead of 46 chromosomes, then they would have they would have triple the number then. Okay, so distinguishing between somatic and germline mutations. So germline mutations, they affect the gametes. So think of germline gametes, right? So germ cells are gametes. So fertilization, so they are going to affect the offspring. They're going to affect the fertilization and subsequent growth into an embryo-form species of a cell in the body. So therefore, when there are changes to the gametes, they're going to be changes to all the cells of the offspring. The offspring inherit muted information, so the information in their genes is going to be mutated, and that can be caused by both external and internal factors. So like we looked at mutations earlier on, it can be caused by external factors, like for instance smoking, if and mammals and stuff. That's why smoking is inhibited during pregnancy, because that can cause it's a chemical mutagen, can cause harm to the offspring. So an example here is that of cystic fibrosis, where there is mutation that affects the gametes. Skin cancer is an example of somatic mutations, where it's only the somatic cells are affected. So it's not going to affect the offspring, it's only going to, or the sex cells, it's only going to affect your already existing differentiated cells. So changes to these will only affect the daughter cells that are produced by mitosis. So it's only going to affect the specific tissue in your body. It's not going to affect the offsprings, and it's usually caused by external mutagens. So with skin cancer, caused by UV rays. So may also be caused during may also be caused by error during replication, and mutations are not passed on to the offspring. So that's important. So that's the difference between germline mutations and somatic mutations. So mutations assess the significance of coding and non-coding DNA segments. So now this module this links directly back to the to transcription, translation, and replication, and what we looked at earlier. So remember, not all parts, as I said earlier when we looked at the promoter and the terminating sequence of during transcription, and I said that not all of the DNA is relevant in protein formation, only some of it is, because not all of it is coding for proteins. So I've got, as you can see, we've got within the DNA, we've got junk DNA, enhancers and silencers, promoters, genes, introns, terminators, and non-coding RNA. So genes, that's a coding DNA, right? Our genes, they code for our external traits. A non-coding DNA is junk DNA, enhancers and silencers, promoters. Introns, we looked earlier how introns are cut out and there are only exons left, and the part that's been coded in mature mRNA when we looked at transcription. So we cut out introns because they don't code for DNA. Terminators and non-coding RNA. So in genes, mutations to a coding region may have effects on what proteins are produced, and therefore that can have effect on cell function. Introns, mutations to introns can have effects on the splicing of the genes. Promoters, mutations to a promoter region can inhibit the ability for a gene to be transcribed. So if there's a mutation to the promoter region, remember where we start the copying, the transcription, if there's a problem there, if there's if there is a mutation over there, that may mean that it is going to inhibit the gene from being transcribed. Terminators, mutation to a terminator sequence may result in incorrect elongated mRNA sequences. Enhancers and silencers, so mutation to these regulator regions that can result in genes being over or underexpressed, so like too much or too little protein being produced, and that is obviously going to have carry-on effects. Non-coding RNA, mutation to coding RNA sequences may result in alterations to important molecules like tRNA. And junk DNA, mutation to junk DNA probably won't matter because it's junk DNA. Cool. So now looking at investigate the causes of genetic variation relating to the process of fertilization, meiosis, and mutation. So we've got mutations, meiosis, and fertilization. How are these now all working together? What impact is then going to have on, especially when we look at fertilization on the offspring? So mutations. Generate new alleles. So point mutations and chromosomal mutations can generate new alleles. So that's where we see variation. There are new alleles, it's variation in the genome. Alleles are recombined and sorted in random, independent, and unexpected ways. These are the ways in which we see genetic variation happen due to the process of fertilization, meiosis, and mutation. So alleles are recombined, sorted in random, independent ways. That happens during crossing over in meiosis, random segregation as well. So we see that the chromosomes are randomly segregated, and independent assortment. Then these variable alleles from these variable alleles from two different organisms are brought together, and they interact in new ways to create new genotypes. And that's when during fertilization these new alleles come together. These genes come together and they give us new alleles. They give us new phenotypic expressions too. So uncontrollable combination of a number of possible gametes, interaction of alleles to produce that new phenotype. So mutations. Now, evaluate the effects of mutations, gene flow, and genetic drift on the gene pool of populations. Again, I know it's a lot going on here today, so I do want you to sort of take it easy in terms of the content, jot down the keywords, and then have a go at, researching them. Have a go, if you want, watch the recording again. But for now, let's go through what each three are. What is the effect of mutations, gene flow, and genetic drift? So we know mutations, permanent alteration to the DNA. We've discussed them, what they are, how they're caused, what is the effect. Gene flow is the transfer of genetic variation, different alleles from one population to another. And genetic drift is when the relative frequency of alleles within a population changes due to disappearance of particular genes as a result of natural selection. So that's when, because of natural selection, we are seeing some alleles from the population drifting away. Their relative frequency decreases because of the changes to a population. That can be environmental changes. They can be natural, like man-made or mammal-made changes, whatever the ecosystem is. So mutations, there's the creation of new alleles of the population. Like we said, mutations, they can be beneficial, but they can also have drastic effect on the mammals as well. Sorry, on the organism. So they can be both good and bad. So it can lead to the creation of new alleles. They may be favorable and subsequently selected. Then gene flow, that changes the frequency of alleles within different population, affects the distribution of genetic diversity. Finally, genetic drift traits become more or less prevalent in the population over time. So you may see a trait becoming dominant over time, and it may lead to evolution of species and creation of new species as well. So mutations, causes of genetic drift. What causes genetic drift? So remember, genetic drift is when the relative frequency of alleles within a population changes because of, due to generally a disappearance of a particular gene as a result of the natural selection. So just opening up my slider again, it's logged me out again, my computers are behaving today, as you can see. Cool. So causes of genetic drift. We first see bottlenecking. When there is an abrupt reduction in the number of individuals in the population, let's say because of, think about like population of like smaller animals and stuff, natural disasters, things happening. When there is that reduction in the number of individuals as a result of sudden and severe selection pressure, natural disasters, that's a selection pressure, or the lack of food because of a natural disaster, something like that. It's severe and it's sudden. That can cause a loss of diversity in the genetic pool. So it's bottlenecking. So you've got the original population, but it's bottlenecking, so only some make it out. So that's the surviving population. Think of the fact that the bottleneck, it's restricted, so not everything, so not all the material in it are making through. The founder effect is when a new population is established by a small number of individuals separated from the larger population. And that will lead to a loss of genetic variation within the new group, and that may lead to new speciation events and evolutionary pathways. A sample of original population, we've got a range of beetles, but they separate. So we've got founding population A, founding population B. So the descendants are primarily A over here, so we see these two types, and then B, these two types. We don't see a mixture, like you don't see any black beetles over here. Okay, so evolution, remember, is a change in the species over many generations. It's not sudden, it takes time. And when we speak about evolution, we are talking about a long process, a long natural process, right? So changes in species over many generations as a result of natural selection of favorable characteristics. So variation occurs within a population due to natural mutation. Environmental pressures are then applied. So we see, physical, chemical competition for resources is a big one. Phenotype best suited to the changed environment, that's going to survive. Remember, the fittest survives, and surviving organisms then reproduce. They pass on their favorable trait to their offsprings, and then gradually that trait is what, that trait is the phenotypic expression of the majority of the population. So a practice question now, let's do it. So the bread mold, Neospora crassa, normally produces its own amino acids from raw materials through a system of enzymes. So we see gene A, gene B, gene C. That produces enzyme A, enzyme B, enzyme C. And then enzyme A, raw materials, so enzyme A added to raw materials will give us the specific enzyme, ornithine. Then enzyme B added to ornithine will give us citrulline, and then enzyme C added to citrulline will give us arginine. So if a mutation occurred in gene B over here, the bread mold will still produce arginine if supplied with what? Think about it for a second, and we'll go through it together. I am a bit hesitant about the time because we've got a bit to go and we're running a bit short on time. So I'm going to go through it quickly because I've got some revision questions on Slido that I also want us to do. I think probably spend a bit extra time on module 5, but that's okay. I think it's more important for you to understand it rather than me just going through everything and not having enough time, and then just having extra time at the end. So, but please keep asking questions. And okay. So hopefully you've had a big think about it and what the question is asking you. So I haven't seen any questions come through yet, but again, if you've got any, please ask. Okay, no questions so far. Cool. So what's happening here? If a mutation occurred in gene B, right? If a mutation occurred in gene B, the bread mold would still produce arginine if supplied with what? And our answer is A. Because if there was a mutation here, it will affect enzyme B, right? And then, even though, you'll sort of have to assume that, even though it isn't said there in the question, but you want to think that, okay, gene B, there's a mutation there, it's going to affect this. That means I won't be able to add enzyme B to ornithine to make citrulline. But if, and I was going to put my hand on the screen and block this whole bit, then I realized you can't see my hand on my screen. But now just look at focus on gene C. If with gene C, which makes enzyme C, all I need is citrulline to add to that enzyme C, these two products to make this. This citrulline has already been made from these earlier processes. I don't have to worry about that. To make arginine, I need citrulline added to enzyme C. So therefore, our answer is A. Have a go at this question. An evolutionary biologist was investigating the timeframes of genetic divergence between different species of acacia. She hypothesized that she would get a better indication of the time at which acacia species diverged by using non-coding DNA segments rather than coding DNA segments. Why is this hypothesis most likely? Why is this hypothesis most likely to be supported by the evidence? Solid 1 minute. Have a go at it, and then I'm going to fly through it and get to the next part. Okay, I've got some good questions. Please keep asking them. All right, so let's go through it. So answer here is D. Non-coding DNA segments will show greater diversity after divergence as they're not exposed to selection pressures. So coding DNA segments never undergo a mutation. That's wrong. Coding DNA because they do, as we've looked through it today. Don't want to be like, it's wrong because I said so, because we've looked through it today. Coding DNA segments are less stable over time due to the selection pressures of the environment. Well, that probably is that one bit where you're like a bit confused between, B and D. Coding DNA segments are less stable over time due to the selection pressures of the environment. True, right? But again, you can think, okay, this kind of seems right, but think to yourself, how does that link back to telling me, how does that sort of link back to telling me about the fact that it's going to give me a better indication of time at which the acacia species diverged, right? How does that link back to the time here? What will that tell me? Yes, they're less stable over the time. They're less stable. They're less stable, but they don't specifically tell me, when there was that point of divergence. Then non-coding DNA segments are stable over time and cannot be used as evidence of genetic divergence. That one we can see it's wrong because firstly you can't assume that it's stable over time, right? Because mutations can affect non-coding DNA and it will, affect phenotypic expressions. When we looked earlier at how non-coding how mutations may affect like the promoter region, how it may affect the introns and stuff, right? So we've looked at that, so we can't really go off that assumption. And then we get to D, which says that known coding DNA segments will show greater diversity after divergence as they're not exposed to selection pressures. And that one is right because it links back to this idea of divergence. It's telling me, it's linking back to divergence. It's showing me something about divergence, which is what I want to know over here. And it's not, and the fact that it's not going to be exposed to selection pressures is also right because changes will happen to the DNA, the coding regions changes will happen everywhere, but it's the changes to the coding region of the DNA that will, face the selection pressure, if that makes sense. So our answer here is D. Okay, this is an 8-marker practice question. So compare the processes and effects of point mutations and chromosomal mutations. Include examples in your answer. As a definition, what are mutations? How do they occur? Point mutations, remember, if they want to start off by talking about mutations, right? It's an 8-marker. You need to have a structure. Then give me the definitions of point mutations and chromosomal mutations. With point mutations, give me a detailed description. What are they? How do they occur? Give me examples of it and how is that? So the process, how they occur, and what effect they're going to have on the cell that we've gone through. Then chromosomal mutations, same thing. Detailed description, examples, how they occur, and the effect they're going to have on the cell. Finally, comparison. This is a compare question. The verb in biology is the key. You need to be able to look at the verb and say, okay, this is what they want me to do. They just don't want me to list what each one is. I can, regurgitate that. But they really want me to now compare the merits of each and talk about, why they're different. What are the similarities and differences? And then give examples for it. So that's that. There is a sample answer there. That's from Nessa, so have a look at that. And yeah, hopefully that will make the structure more clear to you. Biotechnology. Again, I'm going to fly through these. I've got a question. Missed the beginning of this lecture. Can I access the full stream later on? You absolutely can. You can. It will be available from the start. So, biotechnology. How do genetic techniques affect Earth's biodiversity? So, what are social implications and ethical issues of biotech? What are the future directions and what are potential benefits to society? What are the possible changes to the Earth's biodiversity due to genetic changes? Okay, so what is biotechnology? Well, biotechnology, we look at, it's this whole branch of science that uses an application. It's used in medicine, industry, food, energy, environmental sciences, and computation. So according to the UN Convention on Biological Diversity, biotechnology can be described as any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific uses. So we come across applications of biotechnology every day, right? In medicine, in the food that we eat, and in different industries, in the energy that we use. So simply technology that is based on biology. So medical biotech, we look at pharmaceuticals, them producing vaccines, antibiotics, stem cell treatments like skin grafts, treatment of cancers and immune systems, bio-inspired materials like spray on skin, and diagnostics like biosensors. Industrial biotech, so enzyme biotechnology, improving efficiency of industrial processes, biodegradable plastics like biopolymers derived from plants and bacteria, energy sources like biofuels, and photosynthesis. Lots of research surrounding that. And finally, environmental biotech. So we look at bioremediation, so development of microorganisms that are able to clean up pollution. So there's been scientific research around algae being able to do that, and algae being algae being converted to like energy as well. There is also agricultural biotechnology, so genetically modified organisms to better suit changing environments. So a good example of that, I think we get to it, but like Cavendish bananas, research them. They're quite interesting because they have been modified to so that they can avoid a specific type of disease that is popular in bananas. But because all the bananas around the world, Cavendish bananas, are the same, that reduces biodiversity, so now they're more susceptible to that disease. So positives, they meet the growing needs of society. We've got a growing population, so so it's about meeting the development goals like ending poverty and hunger, clean energy, sustainable cities. It harnesses biological tools to invent new creative solutions. So create so and then also it affects genetic diversity, creates new arrangements of genes, new allele combinations. It allows organisms to adapt to their environment, and it's about that. Ultimately contributes to that big idea of evolution. Open source information, so creation of public databases allows for the growing amount of biological knowledge to be shared in a democratic fashion. Concerns here are ownership, right? So should people or corporations be able to own biological information? There may be economic benefits to allow monopolies over biological processes. Intellectual property constraints may become prohibitive to progress. Where is that line between natural and synthetic? There's a huge debate about the food that we eat. Is anything pure anymore, right? Commercial implementation, so biotech monopoly, so companies being able to exploit this information, and being able to use that to gain market control. Consumer rights and choices. Not everyone wants to eat GMOs, genetically modified food. No, so not everyone wants to consume them. So there's that choice, and regulation. So can they strike the balance between safety and innovation, and of course biohacking as a result of this wide variety of information. So, future directions: continual improvement and refinement of current technologies, creation of novel approaches to emerging issues, backyard biotech, synthetic biology, interdisciplinary approach. It's about engineering approach to biological tools, combines molecular biology, genetics, biophysics, and it's about questioning how can we break our knowledge of biology into parts that are then that can then be refined and invented, and it's driven by student-led competitions. So GEM. And you can see where we're giving students the reign to go ahead and being able to work with synthetic biology. So, what are the effects of biotechnology on biodiversity? So, positives: crops may be insect resistant, right? So we may not be able to. So we don't have to use chemicals anymore. Crops may have increased stress resistance and productivity. So that obviously maximizes use of restricted land, grow vegetation where there are harsh environmental pressures, and deliberate introduction of variation. But there are also negatives, right? So ability for GM crops to outcompete, establishment of monoculturing practices. So that's an example where I was talking about Cavendish bananas, where we've just got that one specific type that's going to affect the biodiversity of the species. Horizontal gene transfer into native ecosystems, and competitive advantage may lead to loss of naturally occurring alleles and variation. Evaluations of agricultural practices driven primarily by short-term profit. They have always posed a threat to biodiversity, and biotechnology gives us tools to enact this at a more rapid pace on a more fundamental level. Biotech also has the potential to help the agricultural industry, especially with the shift from urban especially with a shift from rural towns to urban areas, but it needs to be used wisely. Okay, so there's a practice question. We won't have time for this, so I'm going to skip it, but definitely come back to these slides, have a look at them. I'm going to skip ahead because we've still got a bit to go, and time's kind of running out. So bear with me as I go through it. Please pop up questions. Remember, the recording will always be available to you in case you do have to leave sharp at 6. But I'm going to try and get through this and also answer some of your most burning questions. Please ask them. So practice question. Okay, so that is not a practice question. Sorry about the heading, but so specific biotechnology, genetically modified organisms. So when we look at this, you kind of have to consider both the pros and the cons. Utilitarian ethics play a huge role in it, right? GMOs can be used for the good of society. For instance, insect resistance, frost resistance, and it can be used to feed a lot more people, taking into account a growing population. But of course, there are concerns with these technologies, right? There's the concern about the creation of monocultures, horizontal gene transfer, environmental impact. So the essential question is, does the good outweigh the impact? And there is also a question of the balance and the needs. Also, can it be managed rationally? Is there a point where it sort of gets out of hand? And you would see lots and lots of scientific sci-fi movies that sort of show you, the dark side of it. So and that's something to think about, right? So with that, we also have precautionary principle, like if there exists any potential threat of harm, the technology should not be implemented. Companies should prove that there's no significant risk in that product. For example, it should be demonstrated that the technology will not cause environmental impacts. And this is significant considering the high stakes in biotechnology, where risk of significant change to the environment will have impacts on a fundamental level. So infographics, these are some infographics that have been made for our biology classes here at TuteSmart. You can use them, they're here on the slides for you. But like I said, our classes are all about going through the content, giving you like information that's digestible, making it easy for you. So here are some more, helping you convert your knowledge to a band 6. So definitely have a go at these, have a look at these later on. This is such a beautiful poster, like look at this. This is just what we've gone through, but giving you a lot more information. So that's, our content in our classes is tailored to your needs. We're students, we're uni students, we've done the HSC before, we know what in information overload biology is. So these resources are for you. Okay, now last part, genetic technologies. Like I said, we will be going a bit over time. If you need to go, that's all good. I totally understand, but if you can stick around for extra 5-10 minutes, please do. So genetic technologies, we are looking at the uses of current technologies: reproductive technologies, cloning, recombinant DNA, benefits to agricultural medicine, medical and industrial applications, and effects on biodiversity of agricultural biotechs, and influence of social, economic, and cultural contexts. So genetic technology, we've got reproductive technologies, artificial insemination, in vitro fertilization, and artificial pollination. Cloning technologies, whole organism cloning, that's becoming a thing. We're obviously looking at genetically modified crops, but we are also looking at, for instance, mammals. Like there's it was for an assignment a while back, but like camel, one of the camels from Saudi Arabia, there was a clone of the camel, can't remember the name, it had a really cute name, but there's so many examples here. Cow, it's a big one. What's the goat? It's a big example of the sheep, the goat, one of them, I've forgotten it, but you will see lots of examples of whole organism cloning. Please, if you have any questions, please pop them up. I do see a question: what tutoring is available from ATAR Notes? So there is, like I said, group Zoom, that's a tutoring, TuteSmart, and there's also one-on-one private tutoring. But anyways, so whole organism cloning, therapeutic cloning, sorry, that's medicine, and gene cloning as an industry, so medicine and industry. Recombinant DNA technology, so that helps us with gene sequencing, that's helpful for medicine and research. Transgenesis, helpful for agriculture and biotech. Gene therapy for medicine, ELISA, medicine, and CRISPR, so that's a tool, and we'll talk about it. So these technologies, medicine and molecular tool. So Compare the processes and outcomes of reproductive technologies, including but not limited to artificial insemination, artificial pollination, so artificial insemination, injection of semen into the uterus without sexual intercourse, so select animals with favorable trait to breed, synchronized births, avoid injuries, in vitro fertilization, egg is fertilized outside the body, fertility treatments that are used to achieve successful human pregnancies, allows embryo screening prior to implementation. Artificial pollination, that creation of new plants with favorable traits like pure breeding, right, or crossbreeding. You would have looked at Mendel in module 5 and how he talked about the ratio as well with artificial pollination. So artificial insemination, passing on favorable genes to offspring, increased efficacy of livestock industry. However, it may lead to limited genetic variability. In vitro fertilization, artificial pollination. Here is another practice question. Won't have the time to go through it, but please have a go at it yourself. I've got the answers up here. And yeah, and hopefully they help you sort of figure out the structure too. Cloning, so somatic cell nuclear transfer. Dolly the sheep, finally. I was like, there's something there. Yes, someone just posted up there too. Dolly the sheep, thank you. I forgot Dolly. But yes, Dolly the sheep, whole organism cloning. I was confused. I was like, it's a sheep or a goat. But anyways, somatic cell nuclear transfer. So adult cell removed from the organism that you want to clone. The unfertilized egg is removed from the donor and it's denucleated. So you take the so it's empty basically, and the denucleated oh goodness, that's a mouthful. Denucleated egg and the stem cell are fused. The cell is cultured. It begins to divide and become an embryo. An embryo is implanted into a surrogate organism. The surrogate gives birth to an organism, which is then genetically identical to the donor. So the effectiveness here is that offspring may not be strictly identical to parents. Somatic cell mutations, mitochondria, epigenetics, expensive, but it's it's not time efficient, and probs better just to clone, organs rather than whole organism cloning. Gene cloning, so creating lots of copies of a gene of interest, so assembling recombinant DNA to identify the target gene. Remember, we've got the target gene, cut the gene out or order it online, treat gene fragment and plasmid it with restriction enzymes. It creates the sticky ends that are going to stick. Combined sticky ends with will come together by base pairing affinity, and so then we're treating that solution with DNA ligase to repair the backbone. Replicate DNA, insert recombinant plasmid into host bacteria because they'll be super microfactories. Host bacteria will express the gene lots and lots to produce the protein of interest. Alternatively, if your aim is DNA not protein, we also have PCR. So we'll talk about PCR in a second. Alright, so recombinant DNA technology, methods to join together DNA from two different species in order to produce new genetic combinations. So, okay. I don't know what happened there for a second. Sorry, we were far ahead. Where is it? No, it's after Dolly the sheep. Okay, there we go. Okay, so recombinant DNA technology, methods to join together DNA. So we're doing this from two different species. So transgenesis is the introduction of I'm sorry about the noise once again, exogenous genetic material into an organism. So performed, so organism exhibits a new trait. DNA is ubiquitous across organisms. Therefore, we are able to insert new genes into the genomes and code for the new traits. The methods of insertion are this plasmids, so those circular things, so circularized bacterial DNA, retroviral vectors, so transfection, and finally DNA microinjection. So that's a fine glass needle that's going to inject the DNA. How do we make a transgenic organism? Fun fact. So how do we do it? So firstly, identify our genes. Then we get large amounts of those genes of interest and we insert it into a target organism. So gene sequencing allows us to gather that single nucleotide information. We are able to identify the gene. And from this information, we can order genes to then insert into other organisms. Step 2, we use PCR. So PCR, polymerase chain reaction, it amplifies the DNA, so one copy becomes thousands of copies, and so it's used so that gene cloning is more efficient. Then that's technique 2, but there's also technique 3 where we looked at gel electrophoresis. So that's where we're visualizing the DNA and we're checking whether we have the right type of DNA that we need. You don't want to do this whole process and then end up with the wrong type of DNA. Step 3, Restriction enzyme cloning, so we treat the gene of interest plus a bacterial plasmid with the same restriction enzyme. It's going to create those sticky ends, which are then stuck together by DNA ligase, and then that creates a recombinant plasmid with a DNA, which is then transformed into bacteria. So you would have looked at, you will look at, antibacterials. You'll look at the information of antibacterials using this process as well when you study this module. Technique 5 is transfection. It uses a virus as a vector to put a gene into the target cell. Can be used to insert genes into a different cell type, so mammalian, plant. So we place the gene of interest into the viral genome. This is inserted into the organism of interest, and then when the virus affects it. Finally, we are inserting that into our organism. So we do that using DNA microinjection. So just inject the DNA in there. We use a small needle because cells are small. So isolate the DNA. We do DNA sequencing. We identify genome sequences. Creates lots of copies of the genes. Gel electrophoresis, we verify that gene. We put the gene into an organism, and we are able to then transform it. We also do that with viruses, and use transfection as a technique, or we can use microinfections. Either three of them works. So recombinant DNA technologies, we have Bt cottons. We have Bt cottons, and a medical example is the production of insulin in E. coli, which is bacteria. Gene therapy, right? The correction of genetic disorders by the introduction of normal functional cells. By introducing healthy genetic material, the cell and its offspring may have restored function. CRISPR-Cas9, so there are two parts. First part, it guides the RNA containing that nucleotide sequence of the anticodons complementary to the gene that you want to edit. You want to cut out the part that you don't need. And then the Cas9 part will endonuclease enzyme. It cuts the DNA that we need. ELISA is enzyme-linked immunosorbent assay. So a mouthful, an analytical biochemistry tool which is used to detect the presence of antigens in a liquid sample. So diagnostic tool which exploits natural antigens antibody interactions, levels of biomolecules in a sample, so they can be used to measure by linking levels of antigens or antibodies. So it's useful for forensic epidemiology. Another practice question here, unfortunately we won't have the time to go through it, but I've highlighted the key terms for you here. Have a go at it, and then and then match up with my answers over here and see what you were lacking, how you could have improved, or how in that specific sample answer I could have added something else too. Like, it always works in both ways. Agricultural, so we've got techniques and benefits: selective breeding, artificial pollination, transgenesis. Benefits, we get more crops, high yields, more nutritional value. Medicine, we've got cloning, gene sequencing, gene therapy. Benefits are there's personalized medicine, right? Treatment of those genetic diseases, improved diagnostic tools, creation of molecules which can be used for medical treatments like insulin and diabetes. Industry, gene cloning, sequencing, transgenesis. What are the benefits? There's increased speed of chemical reaction, enzymes, creation of organisms which produce industrially significant products, biofuels, biomaterials, energy, etc. So what are the effects on biodiversity? Positives: crops are insect resistant, they don't use harsh chemicals. Negatives, like we discussed earlier, ability for GM crops to outcompete our natural crops. Evaluation, this was the part that I wanted you to think about. Now coming back to it, agricultural practices that are driven primarily by short-term benefit have always posed a threat to biodiversity. With increasing technology, we are seeing this. But biotechnology gives us tools to enact this at a more rapid speed, on a more fundamental level. Biotech has a huge potential to help the agricultural industry, but it needs to be used wisely. So biotech bubble science, there's a huge range of technologies here, and that is something you'll research as you come across it. So what gets made and used is a product of many interlinking factors. Science research is not objective, it's subjective to researchers and fields. And biotech is important, helpful, interesting, and profitable. So I've got examples here. We look at golden rice, BT corn, and also virus-resistant papaya. So what are the social implications, economic and cultural? So this is it here. You can have a look at more resources. My top tips are: understand the fundamental technologies, have examples of them, they're super important. Know your mutations, there are lots of them, different effects, know them in depth. Memorize the examples. You need to know the disease and the disorders and the biotechnologies used. You need to do your research and be critical about it. Finally, write. With this module, you need to be able to give your point and talk about it. So you need to be able to write. So write persuasive paragraphs about different technologies. Exam preparation: consolidate your notes, create module summaries, and do practice papers. Here's an example. Multiple choice questions are very helpful, short answer questions, and long response. That's it from me today. I apologize for going over time.